Thank you all for joining us today. We have over 200 people registered to attend this afternoon, which will be filled with fantastic science. We're thrilled to welcome everyone, and we are also very grateful to our presenters who are going to be providing us with findings as well as advances that they have been afforded by having the opportunity to be awarded an Avant-Garde Award, which is one of our most prestigious awards at NIDA. I'm pleased to have the Deputy Director of NIDA join us for the afternoon and to give some welcoming remarks. And so I don't want to take up any more time so we can get on to our great talk. So Dr. Wilson Compton is the Deputy Director of NIDA, and he also began early in his career as an HIV investigator himself, so I know that the intersection of drug abuse and HIV is a topic that has always been near and dear to his heart, so we're pleased to have him to kick us off with a welcome.
Well, thanks very much, Radana, and I really am glad to be here today. Certainly, understanding and addressing drug use-related risks and the interactions with HIV has been the center of NIDA's HIV research program for over 30 years. And I personally can speak to this because I started my research career on 2 HIV projects that were funded to Washington University in St. Louis to Linda Kotler, and worked with that group for the following 13 years until I joined NIDA in 2002. Now, our HIV research program here at NIDA is the second largest at NIH, and it certainly has made seminal contributions to the science of HIV prevention, treatment, and now HIV elimination. Now, today's symposium is designed to highlight an important component of this research effort, the NIDA Avant-Garde Award Program. Since 2008, the NIDA's Avant-Garde Director's Pioneer, or DP1 Award, has been a flagship initiative of the NIDA HIV research program. This is a highly prestigious award that allows us to fund investigators of exceptional creativity, proposing innovative, high-risk, high-impact research. Now, since its start in 2008, and including our newest 2021 awardees, NIDA has awarded 36 Avant-Garde grants. Today's presenters include, from 2009, Dana Gabuzda, from 2012, Davy Smith, from 2014, Melanie Ott, from 2015, Don Desjarlais and Julie Overbaugh, and from 2016, Stuart Lipton. So we have a wonderful lineup from throughout the history of our Avant-Garde program. Now, please join me in congratulating Drs. Linda Chang and Alex Shalek, who are the newest members of this exclusive group, the NIDA Avant-Garde awardees for 2021. Dr. Chang from the University of Maryland School of Medicine plans to explore the use of MR-guided focused ultrasound as a tool to eradicate CNS viral reservoirs and to promote neurogenesis in the HIV-infected brain. Low-intensity MR-guided focused ultrasound will also be applied to target brain regions involved in addiction for treatment of substance use disorders. Dr. Shalek from the Koch Institute for Integrative Cancer Research at MIT will work to better define the impact of drug use on immune function and fitness against HIV-1. His work will seek to define at unprecedented resolution how substance use disorders influence immune function and response to HIV and other pathogens. He will develop and apply innovative single-cell and bulk profiling and perturbation tools. Now, receiving this award means we are investing particularly in you as a scientist, of course, as well as in your innovative research ideas. So this is a combined award that's focused on the individual as well as the innovative science. Now, let's turn to some of the business for the day. Please join me in thanking those responsible for this symposium, which has a remarkable number of participants. Something over 200 people are watching us today. Our NIDA AIDS research program team includes Radana Chandler, Vasantara Vartakavi, or Kavi, Trishma Smith-Winston, and Andrea Zakowski. Now, our coordinators from LMCI, our support contractor, have also helped us today. And I really want to thank everyone who's assisted in coordinating this event. Kavi and Radana and our contract staff, Aaron Newman, Caitlin Dudevoir, and Jeremy Brundage. It really takes an entire village to pull off a successful Zoom meeting, and I'm very grateful for everyone's effort today. Now, finally, you all may be wondering, how come Wilson Compton's doing this today? Now, of course, my background as an HIV researcher in the past makes me particularly excited to be here today. But the main reason is that Dr. Volkoff is unavailable today. She's with her family this week. It's the first time in over 15 months that she's been able to travel back home to Mexico. And she asked me to cover this event for her, but she particularly wanted you to know how much she appreciates this symposium. She congratulates the new awardees and is disappointed to miss the presentations. She really looks forward to hearing about your findings in the future. And I know both Verdana and I and Kavi will all be speaking to Nora to tell her what you teach us today. So we have to pay close attention so we can be sure to explain it to our director. Now let me turn over the virtual podium to my colleagues from our AIDS research program who will take it from here as we learn about some of the exciting scientific discoveries and the new leads from our NIDA-supported Avant-Garde grantees. I'm not sure whether it's Verdana or Kavi, but I turn it back to you now.
Thank you, Wilson, for that really great welcome and introduction remarks. And I want to also take this opportunity to thank all the panel members, invitees actually, to agreeing to participate in the symposium. I'm really excited and looking forward to your talks. So I'm going to get this kick-started with the first speaker in the lineup, Dr. Stuart Lipton, who comes to us from the Scripps Research Institute. He will be talking to us about TCA cycle compromise due to aberrant protein S nitrosylation in neuroAIDS, methamphetamine use, and neurodegenerative disorders. Thank you. Dr. Lipton.
Trying, hold on. There we go. Great, well, thank you so much for organizing this and for the award and my fellow awardees. It's kind of a life-changing award, let you do what you want to do, and I'll take you through some of that. I'm going to talk today about aberrant protein S nitrosylation. I'll tell you about that post-translational modification and how there are networks of these aberrantly affected proteins, not only in HIV and drug use, but also in other neurologic disorders. And a lot of them are in common, and that's going to be a theme in what I'm going to tell you today. So here we go. As a way of background, approximately 40% of AIDS patients manifest some form of neuroAIDS. The one we mostly study is abbreviated HAND, HIV-associated neurocognitive disorder. The most severe form can result in dementia, although we see a lot less of that with modern CART therapy. Worldwide, some 25 million people use methamphetamine, as you know, and in several studies, as many as 50% in some territories of HIV positive individuals are meth users. So you can see there's a tremendous overlap of these. So what I'm going to tell you about today is that in HAND or neuroAIDS and methamphetamine, there are pathways to neuronal damage that involve in part metabolic insults, and particularly in the mitochondria. Mitochondria are particularly important in producing energy for neurons. We'll talk a little bit about that, as many groups have shown. And then kind of strikingly, the insults that we found in HAND and methamphetamine brains at postmortem, in many respects, resemble damage we've seen in Alzheimer's disease and Alzheimer's disease-related disorders. And this CNS damage is in part due to aberrant post-translational redox reactions that I'll talk about on proteins that involve excessively generated reactive oxygen and reactive nitrogen species that become toxic to neurons and the support cells in the CNS as well. And under the Avant-Garde, I was able to take a new approach to this using a different kind of mass spec analysis to look at these redox altered proteins in human brains, obtained very shortly after postmortem through brain banks here at UCSD and elsewhere, not only in HAND and methamphetamine, but through a series of supplements in AD and ADRD And as I said, strikingly, there's an overlap in some of the major proteins that are aberrantly S-nitrosylated that affect mitochondrial function. I'm going to tell you a little bit of that story right now. So let me tell you what the Avant-Garde has done for me. Not only has it supported publications, the 31 publications during the lifetime of the award. Today, I'm going to really talk about two very recent publications, one in Science, one in Journal of Neurovirology, talking about these mitochondrial deficits that we see and an overlap between hand meth use and an ADRD. I want to shout out to, obviously, the program directors at NIDA have been very, very helpful to me, particularly John Satterley, who's kind of held my hand throughout the award. So this is what we knew kind of coming into the award, 2015, 2016, from our group and many groups. In the CNS, HIV infection came in through infected macrophages and microglia, which produce a series of chemokines and cytokines, gave off neurotoxins, viral proteins such as GP120 and others. They would incite astrocytes that would have aberrant function to affect neurons. So you can have a direct microglial neuronal interaction, but also via astrocytes. We knew that neuronal glutamate would be affected by this, either coming from astrocytes or microglia. That would engage NMDA receptors and other glutamate receptors, increase calcium, and increase reactive nitrogen and oxygen species, particularly reactive nitrogen species such as nitric oxide. Subsequent work has shown that both human microglia and human astrocytes also produce a lot of reactive nitrogen species, although certainly there are other pathways involved. These are major pathways to the damage in neurons. Now, it's interesting because in a whole other set of work done by others, reactive nitrogen species had been, and redox effects of those reactive nitrogen species had been found in meth and other kinds of drug use. Okay, I need to tell you a little bit about reactive nitrogen species and what they do to proteins. So my colleagues and I discovered this post-translational modification about 20 years ago that we've turned, chemists would call it S-nitrosation, we call it S-nitrosylation to kind of make it analogous to phosphorylation. You can also transnitrosylate, that is donating an NO group from one protein to another. And it goes on to a critical cysteine or thiol. Here's the chemistry involved is the protein is R, you deprotonate your thiol. We'll talk a little bit more about the chemical action in a minute. The RS minus reacts with something akin to NO plus, although it's not free NO plus. And you get what I affectionately call a snow protein, NO on the sulfur. It's funny, when I moved to La Jolla from Boston a number of years ago, I thought it would never snow again, but you'll see it's snowing these proteins. And we and our colleagues have published many papers on this, as have others. Now, chemists such as Michael Marletta have looked more carefully at the chemical reaction here. And it's worth taking a minute doing this because you'll see in a subsequent slide, it's really helped us to codify and quantify these reactions in the human brain. So you know nitric oxide is NO dot, an electron in the outer pi molecular orbital, but there are other forms of NO depending on how many electrons are in that outer pi molecular orbital. One reaction mechanism that we think occurs is that ferric heme, or it can be copper, transition metal, can catalyze this nitrosation or nitrosylation. And the way it does it is the electron is accepted by the transition metal. So it's now NO plus and it's donated to a protein producing a snow protein. Another way of doing this, as I mentioned, is transnitrosation or transnitrosylation. In this case, thiolate anion attacks by nucleophilic attack a nitrosyl nitrogen and basically swaps the NO. Here, the NO is on protein number 2, here it's on protein number 1. So we think these are the major reactions that occur in the brain and elsewhere in the body, how you nitrosylate or nitrosate proteins. There are other potential reactions, but we have evidence these are the major ones, and we'll come back to this. Okay, for under the Avant-Garde, we took about 50 human brains, usually with very short postmortem times from brain banks, control brains, brains with hand and methamphetamine. We also did with HIV without hands or without HIV-associated neurologic disease. We homogenized the tissue and went through this series of reactions in order to look for nitrosylated proteins by a new mass spec method, which I'll show you on the next slide. We actually used two methods. First method is an organomercury resonant enrichment method, who my colleague and collaborator Harry Ishiropoulos at the University of Pennsylvania has really pioneered. The second method is called the snow trap method. It uses a triarylphosphine, a chemical that actually traps the NO, so the NO remains there, That was pioneered by chemist Steve Tannenbaum at MIT. Both of these groups we work very closely with. In the first, in the enrichment, the NO was actually removed by reduction by the mercury, and then it's that the protein that had the NO is attached to agarose beads. We can further analyze it. In the trap method, we put the trap and hook it onto biotin, so we have a tag on it. It's now tagged with the NO is actually still there, hooked to the trialphosphine. Then we can submit these for mass spec. We can actually do not only protein identity, but site mapping to see exactly which cysteines are nitrosylated. We can even quantify the degree of nitrosylation of each protein or peptide that we look at. Now, when we do that, this is firstly the PEN method. We use both methods in all of these. I'm going to show you samples. And I can say that the MIT method or the snow trap gives you more nitrosylated proteins. It's a little more sensitive, but many of the proteins overlap with the two methods. So we got several hundred proteins in our first runs in control hand and methamphetamine brains. Interestingly, there's a unique repertoire of aberrantly nitrosylated proteins in hand and methamphetamine compared to control or hand alone, and there are some that overlap. Now, I should say that nitrosylation, like phosphorylation, is also a normal event. Certain proteins are regulated, their functions regulated by nitrosylation. That's what we discovered some 25 years ago, actually, initially on the NMDA type of glutamate receptor. However, with rising levels of NO in the brain during neuroinflammation or other times of insult, then you begin to aberrantly nitrosylate proteins. And that's what we're seeing here when we compare to the controls to the hand meth or HIV brains. Now, strikingly, when you look at GO terms, we found that mitochondrial function was really affected right up near the top of the list, particularly the TCA cycle and respiratory electron transport chain. We'll come back to that in a minute. When we did a string pathway analysis, all the members, all the top hits were in the TCA cycle, interestingly. Okay, let's turn now to Alzheimer's disease. Again, 40 human postmortem brains. Here we were able to do male and female. We had enough of both, and we could use sex and age match controls. This was the snow trap method. Again, we have more proteins, although again, we've done both methods in both. Here we get about 1,500 proteins nitrosylated. It's been estimated from a motif I'll show you in a minute that there may be as many as 12 or 13,000 proteins that are S-nitrosylated. So it rivals phosphorylation, or in fact, might even be more ubiquitous than phosphorylation. Now, strikingly, when we took a look at the Alzheimer's disease male and females compared to the controls and looked at the interactome, again, the very top hits, just like in hand and methamphetamine use, were mitochondrial targets. Mitophagy or autophagy of mitochondria, TCA cycle, respiratory electron transport chain. I don't think this is a coincidence. It looks like there's some commonality between these. So we wanted to study that further. So here's the TCA cycle. And what we've done is look at the effect of nitrosylation on each of the enzymes. Now, under normal conditions, aconitate and isocitrate dehydrogenase are mildly nitrosylated. We think that's normal regulation. Other of the enzymes can be nitrosylated, but the major one nitrosylated in each of these conditions, interestingly, is alpha-ketoglutarate dehydrogenase, shown here. That's the first step that produces NADH, which is the important energy transfer to the electron transport chain into complex 1, so you can make ATP. Neurons are exquisitely poised on disaster. They need a lot of ATP to maintain their synapses. Basal activity of neurons is thought to rely on the TCA cycle, although they can use glucose through glycolysis under certain circumstances during intense stimulation for basal activity. And if you've got a dementia, you obviously have it for years, your basal activity as a neuron is very important. So you need this NADH to go in. And we think that's just, there are other points of NADH production farther down in the TCA cycle, but if you've disrupted here, you can't get there. Now, I won't show you today, but we have evidence from metabolic flux experiments that alpha-ketoglutarate dehydrogenase is definitely compromised. So we came up with a way that we might be able to bypass this. One way would be to supply succinate, the next step, as dimethylsuccinate, which is somewhat permeable, as a kind of prodrug to see if we could bypass the cycle. Now, as you may remember from your biochemistry, complex 2 of the TCA cycle is the same as succinate dehydrogenase in the TCA cycle. So by supplying succinate or a succinate derivative, we're actually not only jump-starting the rest of the TCA cycle, but supplying to complex 2 of the electron transport chain. So we have some evidence that that kind of approach can work. So this uses the Seahorse platform to look. This happens to be iPS neurons from Alzheimer's disease and isogenic controls, but we've also done it on iPSCs insulted with methamphetamine and HIV GP120. Very similar results. What I want you to concentrate on here is oxygen consumption rate, particularly where we look at something called the respiratory reserve. This is when neurons are stressed, how good are they going to do? Are they going to be able to supply energy to the synapses to maintain? We do that by using an uncoupler to see how much energy was there in reserve. And what we see compared to wild type, these are the AD neurons, but as I said, meth or GP120 and so very similar, tremendously compromises your reserve. But if we give dimethylsuccinate, we're not back to normal, but we're statistically protected. Now we're looking to see if we can protect synapses with these kind of rescues, and that work is ongoing. I want to pivot a little bit now and tell you why energy is so important to neurons and other ways that aberrant nitrosylation disrupts it. Really, the synapses, which is the only pathological correlate to dementia in any of these diseases. It's not misfolded proteins, even though they're present and they're important in triggering the events. The number of synapses correlates to how demented you get. And you need a lot of energy, as I mentioned earlier, to maintain these synapses. It's really what makes us uniquely human, why we're having this symposium today. Now, the synapses are lost both in Alzheimer's disease-related dementias, hand and meth use, and other insults to the central nervous system. As I mentioned, neurons particularly are sensitive and need the TCA cycle. So we believe any disease modification of these diseases is going to need synaptic protection. Now, there's another kind of injury to mitochondria, apart from the TCA cycle, that I want to share with you that came as a kind of surprise. As you all know, in the last 15 or 20 years, we've discovered that mitochondria are not these cookie cutter looking little organelles that you learned about in high school. They actually undergo dramatic fission and fusion. And this is an elegantly regulated process by a series of GTPases and their associated proteins. The one that usually causes fission is called DRP1, or dynamin-related protein 1. It's a GTPase that clips the mitochondria. Very regulated. We know these are important in neurological function in particular because there are rare genetic diseases that have a mutation of these GTPases, such as in OPA1, which is a fusion protein giving you a form of dominant optic atrophy. Certain forms of ALS have DRP1 mutations. Charcot-Marie Tooth disease also has mutations. So we know it can affect the brain. Now, what we discovered is that this mitochondrial fission and fusion not only is important in the brain, but it's very susceptible to aberrant nitrosylation. And what we found some years ago initially now is that DRP1, which normally mediates fission, is excessively activated when it's nitrosylated. And what nitrosylates it? Aberrant or oligomerized amyloid beta in Alzheimer's disease, glutamates overstimulating NMDA receptors, HIV and meth insults can do it. They generate a lot of reactive nitrogen species, as I intimated on prior slides. And we get this aberrant nitrosylation on a particular site on DRP1, which causes excessive activation of the GTPase, excessive multimerization of DRP1, and excessive cleavage and fragmentation of the mitochondria, with dramatic bioenergetic failure, resulting in synapse injury. Here's the ATP. We can measure ATP. It falls off quickly when you give an exogenous source of NO, S-nitrosyl-cysteine. If you give GP120 with or without methamphetamine, which makes it worse, or you give A-beta oligomers, all of this cause a reduction of about 50% and a persistent reduction in ATP. Now, we have other ways of looking at nitrosylated proteins. I want to bring this up because the subsequent slide, it becomes important. In addition to our mass spec approach, we have a modified immunoblot that we use that was pioneered by Sam Joffrey in Solomon Snyder's lab a number of years ago using a chemical trick that Jonathan Stamler and I had published. And what we find is that in Alzheimer's brain, but not control brains, we get a lot of nitrosylated DRP1, just like in the meth and HIV brains. Parkinson's disease, not so much, but these are very late stage Parkinson's where you've already lost your substantia nigra. Early in Parkinson's, it may actually occur as well. And the referees made us look at many AD brains. We see it in every brain. There's substantial nitrosylation of DRP1. So by site-directed mutagenesis, we looked at DRP1 and we found one particular cysteine residue at position 644 was nitrosylated. We've produced crystal structure of that with colleagues, and it has what we've called a partial motif that we've discovered that seems to facilitate, it's not required, but it facilitates nitrosylation. And it surrounds that cysteine. So that's probably why that cysteine is susceptible to high levels of NO. Now, what does this do in terms of synaptic damage? Let's look at that on this slide. Spine density, which is kind of one half of the synapse, it's the postsynaptic side. You can actually label it up with GFP or YFP as shown here. These little protuberances are half the synapse. If you expose those to A beta, and many labs have shown this, oligomerized A beta, or HIV and GP120 will do it, or methamphetamine will do it, you lose within a day about half of your synapses. If you block neuronal nitric oxide synthase, you can protect many of them. Now, this is the most important part of this. If you put a point mutation and produce a non-nitrosylable mutant of DRP1, statistically, you totally protect the synapses. So that means that DRP1 was very important in injuring mitochondria, in synaptic failure, and as I said, we can now start to rescue that with bioenergetic compromise. Okay, so the story gets a little bit more complicated. Because of our mass spec methods, we realized that there were a whole series or network of previously unknown nitrosylated proteins that interact with one another. And they're in different biochemical cascades. No one would ever think they ever interacted. So UCHL1, which is a ubiquitin ligase, CDK5, a kinase, DRP1, a GTPase, no one thought they interacted. It turns out they transnitrosylate one another. And we found this either after oligomerized A beta or HIV GP120 methamphetamine insults. And we also found it in human brains with Alzheimer's disease or hand meth, and not in the controls. So let me delve down a little bit on this pathway and show you how we're able to study it. So first of all, we do use mass spec. This is UCHL1. We can tell from these precise molecular weights, masses, we can see exactly what cysteine is nitrosylated because it's altered by the additional weight of NO. And we can identify that cysteine, we can actually quantify it. Again, UCHL1 also has one of those motifs for nitrosylation in the crystal structure that we've looked at. Okay, now going back to the blot method, why would we want to do blots if we have mass spec? Well, I'm going to show you an interesting facet of the blots that we've been able to take advantage of. So we found, for example, UCHL1 not only in HIV methamphetamine brains, but in AD brains, and also CDK5, the downstream, also in AD brains. And we wanted to know how these were related. So to assess transnitrosylation from one protein to the other in a kinetic experiment, we can do this in vitro. What we can do is start out with, we can artificially nitrosylate UCHL1 and then add it to CDK5 and DRP1. And what we see over the course of about 25 minutes is initially CDK5 is nitrosylated, then about 10 minutes later, DRP1 is nitrosylated. If, however, you add to the mix nitrosylated CDK5, the reaction does not go in the other direction. We never see UCHL1 nitrosylated. It's always driven toward DRP1. Now, I realized looking at these blots, we could be much more quantitative because we can quantify the density of the bands. So if you think about it, a nitrosylation is an oxidation. You're losing an electron. We have total protein, which is the reduced and oxidized protein. We can also use a chemical trick to just look at the reduced protein. So now we can look at the oxidized and reduced protein. Why is that important? Well, it turns out that reaction that I had shown you shows you that when you get nitrosylated, you lose an electron. When you get denitrosylated, you gain an electron. Now, someone a lot smarter than I am, named Walter Nernst, discovered that he could quantify this about 150 years ago. And he produced something you all know called the Nernst equation. We use it to look at membrane potential, but that's not why he did it. He was looking at electrons, where electrons went. And I was coming back from a meeting flying at 36,000 feet, probably was oxygen deprived. And I realized, oh my goodness, we could use the Nernst equation to quantify these redox reactions just by quantifying those bands on the block. And when we did that, now we can look thermodynamically and see in a human brain over the course of several years of dementia, what's really going on. And when we plug that in to this modified Nernst equation that I've come up with, which looks at transfer of NO, or an oxidation, from one protein to the other, you get an amazing reaction. So those of you who took physical chemistry will know that a Gibbs free energy can be obtained from the electromotive force of the Nernst equation, minus 18.35 kilojoules per mole. So when you get to minus 20, this is a tremendously driven reaction. It goes to completion. We are driven to destruction by reactive nitrogen species because of this thermodynamic consideration. And so what happens? Well, if you put A beta, you get on IPS cells, for example, you see a lot of formation, as I showed you before, of SNO UCHL1. If you look at iPS-derived neurons from an Alzheimer's disease patient, you lose many synapses. I showed you this before. But if you put in a point mutation in UCHL1, the upstream member now cannot be nitrosylated. You protect the synapses, as shown here and here. Now, we've also done this in vivo using lentiviral vectors. We can put in a non-nitrosylatable form of UCHL1. We've done this in AD models. We're doing it in hand methamphetamine models. And this was actually quantified blindly by Eliezer Meslia, who's now extramural director over at NIA. And what he found is in this presynaptic marker, synaptophysin, the red dye, you can see it's bleached out in an Alzheimer's transgenic mouse made by Leonard Mookie. And if you put in a non-nitrosylatable UCHL1, not only do you totally rescue the number of synapses, you actually rescue normal aging loss of synapses. These mice are about a year old and they start to lose synapses on their own, even more if they have this Alzheimer amyloid precursor protein, and we can rescue that. So what I've shown you in summary then is that A beta oligomers, other misfolded proteins I haven't shown you, but we've looked at HIV insults, methamphetamine, normal aging to some extent, other causes of neuroinflammation, increased reactive oxygen and nitrogen species. They are produced, and I didn't show you this today, but there's a lot of evidence for this in glial cells, both astrocytes and microglia, via iNOS, and neurons via nNOS. These glial cells that are stimulated by HIV produce a lot of glutamate, which engages extrasynaptic NMDA receptors. We'll come back to that in a minute, which actually turns on the neuronal nitric oxide synthase. NO then can nitrosylate TCA enzymes and shut down early events in the TCA cycle, compromising energy. And it can also produce this whole network of nitrosylation. I believe there are many members we still don't know. These are 3 of the members that end up in fragmenting mitochondria, compromising energy, and therefore compromising our synapses and producing cognitive impairment. Now, I want to end on a happy note. The good news is there are ways to intervene. I told you we're trying to intervene at the TCA cycle. That's a work in progress. One way we've been able to intervene, I mentioned this very briefly in the talk, is one of the first nitrosylation sites ever discovered by our group in the early 90s, was the NMDA receptor. This is a normal nitrosylation site that knocks down excessive NMDA receptor activity. We were able to take a drug, memantine, which I had developed and marshaled through the FDA approval process for Alzheimer's disease, attach it to a piece of nitroglycerin, producing a new drug, and we can shut down extrasynaptic NMDA receptors. And to a large degree, that shuts down all these subsequent events. So we think there are ways we can interact and protect these synapses, both from HIV-related and methamphetamine damage, but at the same time, other forms of neurodegeneration, such as Alzheimer's disease and Alzheimer's disease-related dementias. So I've had a tremendous amount of help. I don't have time to mention everyone by name, but both in Boston and in La Jolla, and others have helped me so tremendously. And I am so appreciative to NIDA for giving me this award. And I just want to say that a lot more work remains to be done. And I hope you all come visit us when COVID is over in our beautiful new building for drug discovery at Scripps Research. I'm glad to answer any questions. Thank you.
Thank you, Dr. Lipton. Attendees, if you have questions, please do enter those in the Q&A panel.
Kavi, at least for me, the Q&A panel is not open for questions. So if the logistics contractor can check that and make sure it's open for everyone.
I will check that now.
So Aaron, can someone unmute the participants who have questions? Looks like there are a couple of attendees.
It actually says there's a message to me that you can use the chat to ask questions.
You can use chat as well, whichever works. There you go. It is open. The attendee says it's open. Yeah.
Yeah, we do have a few folks with their hands up. Kumut Singh, we'll go ahead and allow you to unmute to ask your question.
You're still muted.
Sorry, I don't have a question. I think I by mistake, I put the hands on. Thank you. Thanks for a nice presentation. Thank you.
Thank you. How about Paulsami Periyasamy? We'll go ahead and unmute your line.
Oh, it's Shilpa Butch, sorry. Hi, Stuart, excellent talk. So my question is, can you talk about the iron overload and the pool in all these neurodegenerative disorders? Because you showed us the Fenton reaction prior to the nitrosylation.
Exactly, Fenton reaction, but it's related. Yeah, it doesn't have to be iron. It turns out it can be copper in many of these systems too, or any other transition metal. And you don't need a lot of it. And so most neurons have enough of it. As you probably know, ferroptosis, or a form of apoptosis using iron, is very popular now in more acute insults such as stroke. And so we're becoming more and more aware that these transition metals are in neurons. So that's usually not what we, in our view, is rate limiting. Okay.
But do you think that that could be an initiating factor that then goes on to do what you've talked about? Is that one of the key master?
There's a huge argument. The argument's been going on for 150 years of how you get NO onto a protein. And so I'm not going to pretend to know, but we think that is one of the mechanisms. There could be a radical reaction. There's other possible reactions. But once it's on a protein, there's this whole transnitrosylation network. And in a sense, NO has a life of its own. It doesn't need NOS anymore. It's transferring it from protein to protein. And there are at least 3,000 proteins that do this. And this is an untapped, we're about 50 years behind our phosphorylation colleagues, and the chemistry is much more complicated. So we are really ignorant about these networks. We're working on them.
Any of these things affecting the TAT protein, you think? The TAT, viral TAT.
Yeah, so I haven't studied TAT as much as GP120, but TAT, many of these proteins that induce any kind of inflammation will produce a lot of reactive nitrogen species. So the answer is undoubtedly yes.
Yes, thank you. Very good.
There's a question I want to answer in here that in the chat box. Let me answer it. They asked if I did meth alone. No, we were not able to get enough meth human brains to do meth alone. So that's something we'd like to do, but I don't have data on meth alone human brains. We do in a dish. I mean, we've done it and we see kind of additive effects of, for example, GP120 and methamphetamine, but not in the human brain. I don't have that data.
Thank you. There's also a question about, do you see the same sort of effect with cocaine? Or have you tested?
I have not tested. Not personally or scientifically.
Okay. So there's another question. I think, does antioxidant drugs such as NAC might be helpful?
Yeah, so my take on antioxidant drugs is you can't get them to the right place in sufficient quantities. We've taken other approaches to this. The approach we've taken, and it's a whole different talk, but important, is to activate the NRF2 pathway, which is an antioxidant, anti-inflammatory transcriptional pathway, as you all know. And we can do it in cells that are undergoing oxidative or nitrosative stress. And so we have a whole team looking at that kind of approach, and that can be successful. I see another quick question. I don't want to take up too much time, but about synaptic damage key to neuropathogenesis. Absolutely. This is true in every major dementing disease. And we are so tied up in these misfolded proteins, we lose sight that the misfolded proteins do not correlate with how demented you are. And so we've done serial, we meaning Eliezer Maslius and several other, Bob Terry, others have done it. What they've done is serial neuropsychological tests. So we know your cognitive function, postmortem exam, and the only finding at postmortem that we're aware of that correlates with how demented you are is the number of synapses. So we need to protect synapses. That doesn't mean the misfolded proteins or methamphetamine or GP120 isn't triggering the damage, but there's a life of their own. Once you get neuroinflammation going, we need to treat that, we need to protect the synapses. And I think many of us neuroscientists have lost sight of that, unfortunately.
Dr. Lipton, thank you so much. That's all the time we have for the questions. So we'll move on to the next talk by Dr. Dana Gabusta. She comes to us from Harvard Medical School. She'll be talking to us about marijuana use, inflammation, and comorbidities in people with HIV. Dana?
Okay, can you see my screen?
Yes.
Okay, so marijuana use is highly prevalent in people with HIV, with up to 20 to 30% of HIV positive individuals using marijuana for medical or non-medical purposes. Given that marijuana is known to have anti-inflammatory effects, and the fact that HIV infection is associated with chronic inflammation and inflammation-related comorbidities, we wanted to know whether marijuana has anti-inflammatory effects in people with HIV in natural settings or in particular groups of HIV-positive individuals, which could potentially have benefits for inflammation-related complications. We also wanted to know what is the impact of heavy marijuana use, whether beneficial or detrimental, on comorbidities in people with HIV. So in my talk today, I'm going to talk about three aspects of our work. I'm going to talk about our analysis of longitudinal cohort data to understand effects of marijuana on the clinical course and health outcomes in people with HIV. I'll then talk about a new study on smoke-related exposures detected in marijuana compared to tobacco smokers. And then I'll end with a new study on effects of marijuana compared to tobacco on inflammation markers in a large cohort of HIV-positive individuals. In terms of background, it's complicated, and there are many discrepancies between different studies. There are studies that have reported reduced activated immune cells or inflammatory biomarkers associated with marijuana use in HIV-positive individuals. But there are other studies that find no significant association or even modest pro-inflammatory effects, suggesting that these studies in humans, particularly complicated populations such as those with HIV, and trying to understand immunomodulatory effects of, that marijuana might be having in these individuals is very challenging. There are similar challenges in studying the impacts on comorbidities in people with HIV, which to date are still not well defined, with different results reported by different groups. And these results are likely to vary considerably depending on cohort characteristics that affect these outcomes. These questions are significant in order to know can we optimize benefits while reducing risks in people using marijuana for common reasons that are very common in HIV-positive individuals, such as chronic pain, sleep disorders, anxiety, stress, nausea or vomiting, and cancer-related symptoms, or for non-medical reasons. I'd like to begin by highlighting some of the highlights of my Avant-Garde Award, which I received in 2009. These are older studies. We published 18 papers, and I chose 6 to highlight here. We were one of the first to identify a common biomarker signature of immune activation that detected immune activation not only in viremic subjects, but also in aviremic subjects. And these included soluble CD14 and CXCL10, also known as IP10. So we were one of the first to identify IP10 as a very good biomarker of immune activation, including in virally suppressed subjects. We also published a paper characterizing monocyte activation markers in CSF associated with HIV-related neurocognitive disorders, shown here. That also included IP10. And part of our project had an emphasis on using metabolomics and other types of omics and big data to try to understand the pathogenesis of immune dysfunction and comorbidities in HIV-positive individuals with substance use disorders. So these are some of the first metabolomic studies we published. We were the first group to study sort of a global untargeted metabolomics profile in a cohort of HIV-positive individuals, and to find lipid modules shown here that were inversely related to high inflammation markers, bile acid abnormalities indicating liver dysfunction, or to indicators of microbial translocation such as LPS and LBP. And we then went on to study metabolomics profiles in subjects with depression and identified a metabolite profile that included markers of altered tryptophan and dopamine metabolism, as well as mitochondrial energetics in HIV-positive individuals with depression, many of which were chronically using crack cocaine. We then went on to reconstruct the MAX public data set to do longitudinal analysis of substance use disorders in relation to health outcomes. And in a sort of an all by all substance use patterns versus a number of comorbidities, we sort of hit on the ones that were easiest to find first and published one study where we did longitudinal modeling of depressive trajectories in HIV negative and positive individuals, and found a very strong relationship between crack cocaine use and depressive profiles in the HIV positive individuals, much more so than in the HIV negatives. And then we also did a study of marijuana use in relation to cardiovascular events and other comorbidities that I'll be talking more about in my next slides. And one of the most important new directions that resulted from my NIDA Avant-Garde Award was learning how to analyze longitudinal data from large, well-characterized cohorts, including the MAX, the ALIVE, the NNTC, HNRC, and Charter. And we learned a lot of key data strategies that made it possible for us to do more sophisticated modeling of relationships between substance use and disease trajectories and health outcomes. And these are some of the many lessons that we learned here. And we use longitudinal data even for our cross-sectional studies because it provides such an advantage in terms of characterizing levels of use of a substance over time, associations with polysubstance use, and syndemic factors that can also impact outcomes. So this was probably the most risky and difficult thing that I did with my NIDA Avant-Garde Award, which is to basically take the entire MAX cohort public data set from 1984 to 2014 and reconstruct the entire data set into a local SQL so that we could use this for anything we wanted. This was a very challenging project because there's 7,300 subjects followed for like 5 years or longer typically. With 6,000 variables, it was a total of 140,000 visits and over 30 million data points. However, this turned out to be extremely valuable in allowing us to look at multiple different data types and integrate them into new and interesting findings. So one of the first things we looked at was the effect of heavy marijuana use over time on disease progression and health outcomes. We started with a cohort from the MAX, this is from the MAX public data set, of seroconverters, and then another cohort of chronic HIV, which were carefully selected so that at the baseline visit, they had to have a suppressed viral load on ART, and they had to have CD4 counts greater than 300. Another aspect of the study that was important to reach our conclusions was minimizing confounding. And one of the approaches we took was we excluded all HCV positives, and we also excluded other types of heavy drug use, such as crack cocaine and heroin use, to end up with a better matched, cleaner cohort in which we could examine the effects of heavy marijuana use over time on health outcomes. So we developed a technique that we call lasagna plots. Actually, somebody else called it lasagna plots, which allowed us to look at patterns of substance use over time. So shown here on the x-axis is about a 6.5-year follow-up. And then we can code at each visit for each individual subject. So each row here is visit level exposure data for individual subjects, reporting daily marijuana use shown in red, the weekly are shown in orange, occasional use is shown in yellow, less frequent use is like light blue or dark blue. And through this approach, we are able to identify chronic heavy users using daily or weekly for like years. And in this case, we have a cohort where the average use was at least 4 years of heavy use. And we do the same thing for the tobacco users, characterizing the level of heavy smoking relative to non-smoking over time. So this allows us to develop well-defined groups. Another approach we've taken is to look at sort of an all-by-all generalized pairs comparison. I'm not going to go into this in detail, but what this lets you do is take like a big picture view of sort of the all-by-all pairs so that you can not only appreciate differences between groups in marijuana smoking, for example, here, the marijuana users are shown in red and occasionally users in green, and then the non-users in blue. What it lets you do is appreciate differences. And in this case, it's more tobacco smoking and more heavy binge alcohol in the heavy marijuana users. But depressive symptoms were actually fairly similar between groups. We see a depression in CESD, depression scores with age. And it also lets you to get even a little more granular depending how you design this so that you can like look under the hood and see, well, what about people with high education who are also smoking tobacco and marijuana? Are there any differences in their patterns? So we do this for any of our large scale substance use studies. So now that we have our groups, we looked at disease trajectories according to HIV disease markers, viral loads, CD4 count, CD8, and CD4-CD8 ratios. And in both the seroconverters and in the chronic HIV group that we selected, we saw no significant difference by level of marijuana use in any of these HIV-related disease markers. In contrast, we found that tobacco and dual tobacco marijuana smoking in both cohorts was associated with significant elevations in white blood cell counts, predominantly representing expanded neutrophil counts, but still within the normal physiological range, which studies by other groups have demonstrated is an indication of systemic inflammation, even in people without HIV who are chronically smoking tobacco. And this pattern is important because it's been linked to some of the inflammation-related outcomes in tobacco smokers without HIV, including cardiovascular events. So we then did Kaplan-Meier analyses of long-term outcomes by level of marijuana use, and the heavy users are shown in red, the non-users or occasional users in blue. We found no difference in the rates of AIDS diagnoses from between ages 30 to 60, mortality, or cancer diagnoses. And this is important because I'd like to mention we have not found any evidence of association between cancer outcomes and marijuana use in any cohorts we've studied so far. But in contrast, we found a significant increase in rates of cardiovascular events. And this is a merged cohort aged 40 to 60. And this is important because it's easier to detect this pattern in these mostly middle-aged men than it is if you expand the age range, like to go out to age 70 or 75, when other factors commonly have more impact. And these are cardiovascular events, either by just tobacco smoking, with these tobacco smokers shown in yellow, or a three-way stratification by marijuana only, shown in green, or marijuana plus tobacco, shown in red, showing these effects were detectable in the HIV positive individuals, but were not significant in the HIV negative counterparts, possibly suggesting that the already elevated risk of cardiovascular disease in HIV positive individuals may bring out the added effect of marijuana and tobacco smoking on these outcomes. These are adjusted logistic regression models showing that in adjusted models, including other risk factors, viral load, and CD4 counts and age, we detected about a twofold odds ratio in either marijuana users or cigarette smokers for cardiovascular events in middle-aged men in the MAX. These studies suggest that heavy marijuana smoking, and when I say heavy, I'm thinking about those red lasagna plots with 4+ years of daily or several times a week use, is associated with an increased rate of cardiovascular events in HIV-positive men ages 40 to 60, beginning with some events occurring at relatively young ages, including myocardial infarctions or strokes under age 50. This may explain a portion of the elevated cardiovascular risk in some HIV-positive populations that is not attributable to tobacco smoking and traditional risk factors. We then went on to do a similar type of study using the MAX public data set, and this time we took an even wider time range from 1996 to 2014, where we compared the effects of marijuana smoking on pulmonary disease in HIV-infected and uninfected men. We did a matched group design where we first excluded other types of heavy drug use. And then we used a match software to identify matched groups of HIV negative and positive participants matched for age and race at the baseline visit. And in this study, marijuana smoking was defined of at least 1 year of daily or weekly use, and we used a time updated variable with a sliding window of a 2-year average to identify heavy marijuana or tobacco smoking over time. The major findings from this study were a significant impact of heavy marijuana use, shown in blue, tobacco use, shown in yellow, or the combination of using both marijuana and tobacco on self-reported chronic bronchitis in HIV-positive individuals, or on a composite of infectious pulmonary diagnoses that included viral pneumonia, influenza, bacterial pneumonia, quote, other causes of pneumonia, which presumably was mycoplasma. And that was associated also with an effect of marijuana on these, on a composite of infectious pulmonary outcomes between ages 30 to 65. However, we did not find similar findings in the HIV negative group matched for similar age and race at baseline. And also, we did not find associations with other pulmonary outcomes, including COPD or lung cancer in terms of the marijuana exposure. We then wanted to know more about the mechanisms through which marijuana smoking, or heavy marijuana smoking in particular, might lead to elevated rates of cardiovascular and pulmonary comorbidities. And to accomplish this, we performed a study in collaboration with the Tobacco and Volatiles Branch Group at the CDC, in which we measured 33 plasma and 28 urine metabolites of nicotine, THC, and various smoke exposure related chemicals such as polycyclic aromatic hydrocarbons, VOCs, and other smoke related chemicals using mass spectrometry. This was done in a longitudinal cohort study of 245 participants all over age 40. 76% were HIV positive, and the subjects were enrolled mostly in the NNTC cohort or the HNRC and charter cohorts, and we had about 40 subjects included from the Chicago site of the MAX. We then evaluated these findings in relation to exposures and health outcomes. This is the overall characteristics. The median age at baseline was 53. The groups were pretty well matched for age. Most of the characteristics are fairly well matched, but interestingly, there were more cardiovascular diagnoses within 5 years prior to endpoint, particularly in the tobacco smokers and to a lesser extent in the marijuana users. So these are the lists of the metabolites that were measured in plasma and urine. We compared levels both by geometric means and t-tests, as well as in these box plots with non-parametric tests. And shown here are the most notable findings. So the 4 groups are shown in different colors, where blue are the non-smokers, the green are the marijuana-only smokers, yellow are the tobacco-only smokers, and the red are the dual users. And as shown here, we detected good levels of THC in just about all of the marijuana users, or in the dual users who are both using marijuana and tobacco. And the marijuana users had essentially no nicotine metabolites, so they were clean, and that was partly by our study design. And then we looked at the other metabolites, and the significant findings in terms of marijuana exposure were that marijuana smoking was associated with elevated levels of plasma naphthalines. Here's one example shown here, as well as other metabolites, including O-cresol sulfate. But most notably were the acrylamide and acrylamide shown here, AMA and gamma, and acrylonitrile metabolites, SIMA and SIHA, which were elevated in marijuana smokers who are not using tobacco, shown here in green compared to the non-smokers. But these elevations were generally not as great as those seen in tobacco smokers. And this was generally true across the board, that the highest levels were seen in the tobacco smokers or the dual users, with relatively modest elevations seen in the marijuana-only users. Now, contrary to our original hypothesis, we did not find additional elevations in the dual users who were smoking both marijuana and tobacco. Their levels were relatively similar to those of the tobacco-only smokers. And these are heat maps of the same data organized by group, the non-smokers, the tobacco-only smokers, these are the marijuana-only smokers in green with the THC and then low levels of some of these smoke-related chemicals. These are the tobacco-only smokers with high levels of these smoke-related chemicals. And then these are the dual smokers with similarly high levels of these chemicals. And I wanted to bring your attention to HPMA, which is the major metabolite of acrolein, which is considered one of the most toxic chemicals in tobacco smoke, and show you here that acrolein was high in the smokers, only detected in a few marijuana smokers, but at levels that were really no different from the non-smoking controls. And you may wonder, where's this acrolein coming from in these non-smokers? Well, it's because there are other sources, particularly dietary sources related to fried foods and charred meat. And acrolein is thought to be one of the major reasons why fried foods are unhealthy for cardiovascular risk, because it is a reactive aldehyde that the major source is derived from combustion of tobacco and other combustible products, but it can also be derived from food sources, especially fatty foods. And it's reactive, so it can cause reactive modifications of proteins, for example, in vessel walls. And it can also activate neutrophils and monocytes by its reactivity. And it's been associated, we and others have associated with tobacco smoking, shown here the correlations with nicotine metabolites. And in adjusted models, we associated high acrolein levels with cardiovascular risk adjusting for other factors, either in the total cohort or just in the HIV positive subset.
Dr. Gabbuza,
Yes.
a few more minutes before the Q&A.
Okay, thank you. So anyhow, this shows that acrolein exposure is increased by tobacco smoking, but not exclusive marijuana smoking, and contributes to cardiovascular disease in tobacco smokers. Since I'm running short on time, I'll just briefly talk about our profiling of inflammation markers in a cohort of 317 individuals. 232 had HIV. They were from NNTC, Charter, and HNRC, and 95% of them had plasma viral load suppressed below 200 copies. The marijuana users were all using for at least 1 year, and they had detectable THC in urine or plasma at the sample visit. This is another G-pairs plot, and I'll basically give you the punchline since I'm running short on time. When we looked overall at the total cohort, we really did not see any significant effects of marijuana use on inflammation markers such as gamma interferon, IL-1, IL-6, and CRP. However, we did detect an anti-inflammatory effect on these markers if we focused on the HCV positive subset. And it's well known that co-infection with HCV further augments inflammation levels in HIV positive individuals. And in these individuals, we could detect an anti-inflammatory effect of marijuana use on inflammation markers. And we looked at this in adjusted models, both logistic regressions and linear regressions, where marijuana's effects alone were not remarkable. But when we included an interaction term of marijuana with HCV serostatus, we could detect an inhibitory or negative effect of marijuana use on the levels of these three inflammation markers, interferon gamma, IL-6, and CRP, which had the strongest findings on suggesting that you need to look at these cohorts very carefully in terms of context. And this is a population at greater risk of inflammation. And in this subgroup, we can detect anti-inflammatory effects. So to conclude, our studies, together with findings from other studies in humans, suggest that the anti-inflammatory effects of marijuana in people with HIV are context dependent. Modest effects on inflammation markers, particularly IL-6 and CRP, are detectable in some natural settings where inflammatory activity is high, such as HCV co-infections, when the study design accounts for covariates such as tobacco smoking, BMI, viral suppression, etc. We've also shown that long-term heavy marijuana use is associated with elevated rates of cardiovascular events, infectious pulmonary diagnoses, and chronic bronchitis in men with HIV independent of tobacco smoking. In contrast, however, we did not detect significant associations of heavy marijuana use with rates of progression to AIDS, mortality, cancer, COPD, or cognitive impairment. We've also shown that marijuana smoking is associated with exposure to some of the smoke-related chemicals that are also associated with other types of smoking, including acrylonitrile and acrylamide metabolites, but the exposures are lower in comparison to tobacco smoking, and the health risks of these exposures remain unclear. So in sum, given these findings, the high prevalence of marijuana use and increasing legalization, there is an urgent need for well-designed research to better understand potential benefits and risks of various marijuana products with different potencies and different routes of deliveries and different ratios of CBD to THC, which might have different effects on inflammatory profiles, and to determine the safest routes of delivery. These are the people in my lab who did the work, particularly David Lawrence, Vikas Misra, and Deb Jani, who did most of the work that I'll present today. And these are our collaborators at the NNTC, the MACS, and the CDC. And I'd like to end by thanking you for your attention and be happy to answer any questions.
Dana, that was a beautiful talk. Thank you very much. We have some questions that have come in. So do you have any insights on the differential impact of smoked versus vaped cannabis?
I couldn't hear you, I'm sorry.
Do you have any insights or data on differential impact of smoked versus vaped?
That is a very important question. We would love to have been able to dissect our data in terms of the smoking versus the vaping versus different ways of smoking, whether it's a joint or a pipe or a water pipe, et cetera. As we suspect, there probably are differences, but we did not have the data available to do this. And one thing I would like to encourage is for these well-characterized observational cohorts to get some kind of additional funding for new questionnaires that could be added on to these existing cohorts so that we can collect that data, not only about the route, the smoked versus the vaped, but what products are they using? What kind of strain? Is it the high CBD variants? And, I think that would be incredibly very useful data because I suspect we will find differences, but right now there is virtually no data to address any of those questions for marijuana use.
So we can tell you from a program perspective, the NIDA program staff are working with the NICHD, now managing MaxWise cohort, in trying to introduce some of these instruments to get more clarity on types of drug use and the modes of use. We are met with a little bit of pushback because already the visits are so long with so many different instruments that the participants have to take, but we are pushing for it. So hopefully it will happen pretty soon. We have more questions here. Okay, so were you able to look at any data comparing other routes of marijuana, which you just answered?
On pulmonary disease, that's one of the first questions. Yeah. Other ways of smoking, does that affect pulmonary disease? I suspect there will be differences between the smoked, the vaped, the non-inhaled routes, that there will be differences. But right now, I don't have any data to address this, and I'm not aware of even data in the general population to address that question. I think those studies are awaited.
Any data by gender? Like, what are the consequences in women versus?
That's a good question. In the HIV positive cohorts, we tried to look at that, but we did not have enough females to say whether there's a difference. But in many studies, females are more prone to high levels of inflammation, especially if it's combined with female with high BMI and other risk factors or lifestyle factors. But so, you know, it would be very interesting to look at that question, but we didn't have the statistical power to be able to do that. I'm not aware of data from other cohorts that have strongly addressed that question, but it's a good question.
Right. We have.
Oh yeah, so I'm going to answer the last one because that's an
Yeah.
easy one. So anonymous attendee, do you find IL-6 has an exclusive pro-inflammatory impact in your work? We did not yet try to tie IL-6 data to what is the impact. Like, do people with higher IL-6 in our cohort of 300 people have more cardiovascular disease or more of anything else? We would like to do that, but we haven't done that yet. And given all the confounders that I showed you in those giant grids, those will need to be carefully adjusted models to be sure that the outcome is related to one factor and not another. But I would like to emphasize that of all the biomarkers we looked at, IL-6 and CRP seem to be the ones that had, you know, the best signal to noise, and I think are ones that we're going to continue to focus on. IL-6 is also interesting because of the so-called inflammaging story. And we also see trends of higher IL-6 with aging and CRP too. So those are good biomarkers to look for a number of outcomes, including sort of frailty and aging related effects.
That was part of my question I was going to ask, but
Yeah.
Dan, are you looking at COVID?
Oh, so I'm not looking at COVID, and I knew somebody was going to ask about COVID. So I looked at the literature, including I even looked into the BioRx unpublished stuff that's posted on, has anybody looked at marijuana smoking or vaping to say whether there's more risk of bad pulmonary outcomes in COVID? I cannot find any real data that's looked at that. And I think most of the studies have looked at tobacco smoking and vaping, and yes, there is a higher risk for COVID-related outcomes, and that's why they got prioritized for vaccine groups in some of the states. But for marijuana use, I can't find any data on that, and I think it's a very important question. I think a lot of these hospital-based and EHR-based databases just didn't have enough data for the people doing the studies to be able to quickly answer those questions.
The paper in molecular psychiatry that Dr. Volkoff is co-author on, and there's a companion paper in World Psychiatry, addresses cannabis use disorder in the huge IBM data set. So that's worth considering.
Thank you. Thank you very much. Yeah.
And it does show an association with poor outcomes. It's kind of universal for all the substance use disorders. It's an association with COVID-19 diagnoses.
Okay.
Opioid use disorders at the top, but virtually all of them are significant risk factors, adjusting for multiple other possibilities.
Okay, so I'm dying to answer this question from Monique Brown because it's really important. I am surprised there was no effect on cognitive impairment. Does marijuana use usually affect cognitive impairment? So we have looked at cognitive data in almost every cohort that I mentioned, because a lot of them have a focus on cognitive outcomes, and you can get T-scores and depression scores, and you can get diagnoses of the so-called hand-related diseases that Stuart Lipton was talking about, the ANI and MND, you know, different types of cognitive outcomes. And to date, we have not seen either a positive or negative relationship between heavy marijuana use and either T-score data or hand-related diseases. We haven't really drilled down to start looking prospectively or like 5 years later or in an aging cohort. But I think it's an important question because different cohorts report different results. So Ron Ellis in Southern California has found potentially beneficial effects, and Igor Grant as well, saying that marijuana use was associated with lower incidence of HIV-related cognitive disorders in their cohorts. And then there are studies on the East Coast that have said the opposite and have said that they find higher rates of cognitive impairment, particularly memory disorders, in people who are reporting heavy marijuana use. And I think part of this is exactly like one of the attendees that just said, depends how much you use, routes of exposure, other cofactors like tobacco smoking or not, and potentially even the products that are available in the regions where the studies are being done. You know, the products on the Southern California might be different from what people are getting either on the streets or in the dispensaries in other states. So I think it's very complicated. And I think the different studies in their context could be correct, but they need to be viewed in the context and the setting and the products that are being studied in that study. So we have not detected positive or negative effects, but I think we'll stay tuned and keep looking. And these are not, somebody said, you would think there would be impairment, but when we study people at the study visits, we assume most of them are not coming in very impaired from any recent drug use. Otherwise, they might not be coming in to do the entire neurocognitive battery. So I think when they're looking in the neurocognitive batteries, presumably they're mostly looking at the sort of more long-term effects rather than the effect of an acute exposure. But anyhow, remains to be determined.
Thank you, Dana. That was a wonderful talk, and I think that's all the time we have for questions.
Thank you.
But feel free to answer them if you can in the Q&A, if you have time. And let's move on to the next talk by Dr. Don DeZorles. He's from School of Global Public Health, New York University. He will be talking to us about combined prevention of initiation into injecting drug use. It is, trying to find, okay, there you go. Don, you're on. Do we have to man the slides, or are you okay with?
My medical center put a very heavy firewall on my computer, which does not allow me to do any screen sharing. There's great concern about the confidentiality of data within the medical center. So I do have that problem. Okay, so
These are great. Yeah.
if we could go back to one slide. Okay. So I'll be talking about reducing initiation and relapse into injecting drug use. Next slide, please. Okay, I'd like to acknowledge the team members from New York City and also from Tallinn, Estonia. This has been a long-standing international collaboration looking at HIV among people who inject drugs. In New York City, we were basically studying a suburban opioid epidemic. And in Eastern Europe, we were studying an HIV epidemic, a long-standing HIV epidemic among people who inject drugs. Next slide, please. Okay, the problem of injecting drug use is becoming universal throughout the world. It's a very cost-effective method of administering drugs. You get a very intense drug effect at low cost because you're using almost all of the drug. Even compared to smoking, you use most of the drug and get an intense effect. And that's an immediate effect. So in terms of habit formation and behavior change, an immediate effect can be quite powerful. There are, however, many adverse individual and societal consequences of injecting drug use, which everybody is familiar with: bloodborne viruses, bacterial infections, increased likelihood of overdose, and more rapid development of dependence and substance use disorders. These, however, tend to be delayed effects. They may take months to even years to development. So again, in terms of setting up habits and behavior modification, the delayed effects are likely to have less importance to a person who is contemplating beginning to inject drug use. Next slide, please. Okay. In this avant-garde study, we use the logic of combined prevention. That's similar to the logic of combined ART treatment for HIV infection. You don't use just one drug, you use a combination of drugs to get maximal effect. It's also the same logic in preventing HIV infection among people who use drugs. that when you use multiple interventions, such as needle syringe programs, ART, and medication-assisted treatment, you can see actual elimination of HIV in population of people who inject drugs. For the problem of beginning injecting drug use, we looked at programs to reduce initiation of new injectors by current injectors, programs to keep non-injecting drug users from seeking a first injection, programs to prevent relapse by injectors who have changed to non-injecting drug use. That's an interesting thing that has come up several times in the study, that many people who inject will decide they don't want to inject anymore, and they'll change to non-injecting forms of heroin or cocaine use. And the problem then is to keep them in the non-injecting mode of administration rather than relapsing back to injecting. And we also looked at medication for opioid use disorders, because if you can put somebody on buprenorphine or methadone, you can dramatically reduce, possibly even eliminate their injecting, eliminate the adverse consequences of injecting. And if you can get people onto mood before they start injecting, it may prevent initiation of injecting. Next slide, please. Okay. I mentioned that we did this in New York City and Tallinn, Estonia. In New York City, we worked mostly on Staten Island, which is the suburban borough of New York. It was in the midst of an opioid epidemic, of people going from painkillers to heroin and from non-injecting to injecting drug use. It had very, very high rates of non-injecting drug use, and heroin was the primary drug for both non-injecting and injecting use. Tallinn, first, Estonia is a Baltic country near Russia. Tallinn is the major city. It is towards the end of an injecting drug use epidemic. Their injecting drug use epidemic started with the collapse of the Soviet Union, and they got up to HIV rates of 50% or more. They have low to moderate rates of non-injecting drug use. Interestingly, fentanyl and amphetamine are the primary drugs used. Fentanyl came into Tallinn and basically drove out heroin. So it is the place in the world where fentanyl has been used the most intensively and for the longest period of time. It has traditionally had the highest rates of fatal overdoses of any place in the world. And the avant-garde has allowed us to continue to study a fentanyl epidemic. Next slide, please. Okay. Illicit drug injection is a complicated, messy, and dangerous procedure. It's potentially fatal, and almost everyone requires the assistance of an experienced injector for their first injection. It's not something that you just pick up by yourself. You typically would never have the skills to just start injecting by yourself. Next slide, please. Okay, this is a picture of somebody giving an injectant to someone else, and I'll focus on both people in this situation, the person receiving an injection and the person giving an injection. Okay. Next slide, please. This avant-garde was built on some previous research, particularly the heroin sniffer project that we did in the 1980s. That was an intervention for heroin sniffers, intranasal heroin user, both people who had never injected and people who had formerly injected, they had stopped injecting. It was a small group intervention with trained group facilitators. It focused on providing information, motivation, and modeling of behavior. And it was a randomized clinical trial. Next slide, please. We did an 8th-month follow-up. We found that in the experimental group, 15% had injected during follow-up, and in the control group, 33% had injected during follow-up. So an odds ratio of better than 2 in terms of participating in the study, reducing the chances that you would inject drugs during follow-up. Injecting during follow-up was associated with the recency of last injection among the former injectors. If they had injected within 6 months of coming in to the study, they were much, much more likely, an odds ratio of about 4, to inject during follow-up. And it was also associated with close personal relationships with current injectors, that the more friends or family or sexual partners you had who were injecting, the more likely you were to inject during the follow-up period. Next slide, please. We adapted this intervention into what we call avoid the needle. We changed to one-on-one sessions with a booster 1 month later. If you've tried to work with people who inject drugs, getting them together for group sessions is a major logistical challenge, so we moved to one-on-one sessions. We had slightly different versions for the people who had never injected and for people who had formerly injected. We focused on motivational interviewing to reduce ambivalence about injecting, emphasize concerns about immediate negative consequences rather than long-term consequences. It was a strength-based intervention where we tried to develop their particular skills, and we gave them skill practice in refusing offers to inject. We also provided information on naloxone, where they could obtain it, given the overdose epidemic going on in both locations. And we provided information about safer injecting in case they did start injecting. Next slide, please. The components of avoid the needle is, first, there was an introduction. Then we reviewed people's current drug use and their history for their main drug. And we got them to think about 1 year in the future. Most people who use drugs think about today and tomorrow, and that's about it. So we tried to create a longer time frame for thinking about their drug use. We've reviewed their knowledge of injecting, and many of the never injectors were pretty ignorant about injecting, their thoughts, feelings, and have they ever been in situations where they were close to injecting. We reviewed the health, social, emotional, and legal risks of injecting, and discussed how to handle a situation where injecting may be imminent. Next slide, please. We did role plays of refusing to inject in the face of social pressure from other people to inject. And we found that that was particularly useful to give them skills in refusing to inject. We also, as I mentioned, discussed safer injection and dealing with overdose. And then we had a wrap up of what they would take away from the session. Next slide, please. We recruited from SSPs in Tallinn in Staten Island. We had a pre-post design with a 6-month follow-up. We had the same pattern of results that we had in heroin sniffer, a very, very low rate, under 15% of people who had never injected, starting to inject, a very, very low rate of relapse among the long-term former injectors, but a high rate of relapse among the short-term former injectors. That if your previous last injection was within 6 months, you were very likely to go back to injecting during follow-up. But if you'd gotten past that 6-month period, people tended to form a stable pattern of non-injecting drug use with very, very low chance of relapsing back to injecting. And we found that it was concerns with friends and relatives' reactions to going back to or starting to inject that was the most important factor in whether or not they did inject during or avoided injecting during the follow-up period. Next slide, please. We did have a major finding that it was very difficult to recruit non-injecting drug users at organizations. The non-injectors did not want to come to the syringe service programs. They wanted to maintain confidentiality of their drug use. They didn't want other people to know that they might start injecting. Concern about track marks was an important factor in not injecting. We also tried working with schools, drug treatment programs, and drug courts, and found that those organizations were basically not cooperative. They wanted to deny that any of the people they were working with used drugs. And if they admitted that people were using drugs, they preferred an abstinence-only approach rather than a harm reduction orientation. That they wanted the people that they were working with to just totally stay away from drugs and not consider safer versus more dangerous types of drug use, sniffing heroin versus injecting. Next slide, please. We then restarted this study in New York City with street-based respondent-driven sampling and found many non-injectors were willing to come to an anonymous building. It was actually part of the dental school. So that if they could come to participate in the study without risking their confidentiality, we had no problems recruiting non-injecting drug users. Then COVID hit, and as part of the lockdown in New York, we had to shut the study down. We were not allowed to see research subjects in person. Months later, we were able to restart the study based on telephone interviewing. We mailed payment cards to subjects so that we could load money onto a Visa card after we interviewed them. But unfortunately, many subjects had moved. They changed phone numbers. Some had moved from various shelters due to COVID. But we formed a new hypothesis that the COVID pandemic conditions would accelerate transitions into and relapses into injecting drug use, that the stress of the pandemic would increase the likelihood that they would move on to injecting drug use. Next slide, please. But in the data we've collected so far, that hypothesis was not confirmed. None of the 42 subjects we've interviewed so far injected during follow-up, and we would have expected from our previous research that 7 or more of them would have injected. So we had a clear rejection of the hypothesis that the pandemic would lead to worse forms of drug use. We started thinking about resilience among non-injecting drug users. A number of them told us that they had taken the pandemic as a time to re-examine their drug use and trying to reduce their drug use and definitely staying with non-injecting drug use rather than moving on to injecting. As a side note, the COVID vaccination rate in this group was approximately equal to the rate in New York City as a whole. Next slide, please. Next, I'll talk about the person actually giving the assistance with the first injection. There was previous research on an intervention called Break the Cycle. It was originally developed by Neil Hunt in the United Kingdom. A second version using peers or people who inject rather than trained counselors was developed by Carol Strike in Toronto. It's based primarily in motivational interviewing, and the studies have seen relatively large effect sizes with 50% or greater reductions in having people who are currently injecting assisting with first injections in pre versus post designs. Next slide, please. We wanted to update, break the cycle. We first conducted qualitative studies of people currently injecting drugs in New York City. The qualitative studies gave us 4 different types of PWID with respect to assisting with first injection. There was a group that we called the Kantian moral imperatives. They just never, ever wanted to assist with the first injection. That was the large majority of people who were currently injecting. We also found a group we called the situationalists. They generally do not assist, but they will assist if they believe that that will cause lesser harm to the non-injecting user who wants the first injection. We found a group we called the opportunist, who would assist for drugs or other considerations. And we found a small but worrisome group that we called the evangelical assisters. They were people who believed that injecting is by far the best way to use drugs, and they wanted to turn on people who were not currently injecting. Again, that was a small group, but definitely a worrisome group. Next slide, please. Okay. We used a clinical trial research design for our updated test of break the cycle. Next slide, please.
Doctor, just a few more moments before the Q&A, please.
Okay, I will go through quickly. We used subjects. Okay, next slide, I'll go on to. Okay, it was a pre-post design, 6-month time periods. Next slide, please. I'll skip over the components of the updated intervention. I've mentioned it's primarily motivational interviewing and skills training. Do want to note that we did a role play of refusing to ask if they were refusing to assist if they were asked. Next slide, please. People often regretted their first injection, often with intense emotion. Next slide, please. These subjects were very different in the 2 cities, particularly in their drug use and their ethnicity. Next slide, please. Okay. We did see very different patterns of injecting drug use. As I mentioned, it was fentanyl and amphetamines in Tallinn and heroin and cocaine in New York. Next slide, please. We looked at injection promoting behavior, whether or not people talked positively about injecting or modeled injecting in front of non-injectors or offered to assist. Next slide, please. Okay, these were the outcomes. In Tallinn, we saw a reduction in promoting behavior. And in both sites, we saw substantial reductions in assisting with first injections, an 80% reduction in Tallinn and a 60% reduction in Staten Island. Next slide, please. In red, we looked at the baseline to follow up for the people who received the intervention and noticed that we saw reductions in their own injecting, reductions in non-injecting drug use, statistically significant. Next slide, please. So for additional studies, we've done another round of break the cycle in Tallinn, and we found equivalent results of 80% reduction, but evidence that the intervention was not sustained over a 2-year period. We were doing an RCT to break the cycle in New York to quantify reductions in assisting and reductions in people's own drug use. That did get interrupted by the COVID pandemic also. We were doing an implementation of break the cycle at an SSP where we would be able to sustain it because the people doing the intervention would be staff members, and they would get reimbursed for substance use counseling. Again, that was interrupted because of the COVID pandemic. Next slide, please. So in terms of our next step, we really want to get everyone vaccinated for SARS-CoV. It is very, very, very difficult to do interpersonal interventions when COVID is not under control. Next slide, please. We want to strengthen the break the cycle intervention for reducing injection promoting behavior, further study the effects of break the cycle on participants' own drug behaviors, try to identify the active ingredients in break the cycle, and examine break the cycle for sustainability and the possible need for booster sessions. Next slide, please. For avoid the needle, we wanted increase the focus on interpersonal relationships because those seem to be critical for whether or not people start or relapse to injecting. We need to find institutional homes for providing avoid the needle. We don't really have an infrastructure for providing this intervention. We want to develop a more specific intervention for people who've recently transitioned from injecting. If we can get them to 6 or more months of non-injecting drug use, we feel they're very, very unlikely to relapse to injection. And we want to try to increase access to low threshold for mood. That has happened because of COVID, and we want to see if we can continue that. And next slide, please. And thinking at a bigger level, we're considering working with PWID in Haiphong, Vietnam. We recently completed a study there where HIV incidence has been reduced to less than 0.1 per 100 person years at risk, essentially eliminating HIV among people who inject in the city. But 45% of these people who inject heroin are smoking amphetamine, and about 5% have injected. So there's a potential massive spread of injecting methamphetamine among these people who are currently smoking. We're thinking in terms of population level implementation of combined interventions to prevent transition from smoking to injecting methamphetamine. I think that this would really need to be done at a population level, that you can't really prevent epidemics of injecting by working with small groups. But if we can develop methods for preventing injection of methamphetamine, that would have great implications for the US as well. Okay, thank you.
Don, thank you so much. That's a very interesting talk. So I'm trying to, so we kind of like went over time.
Yeah, I was afraid of that.
That's fine. So if you could, please go ahead and answer any questions that might come up in the Q&A.
Okay.
Because it's so hard to hang on the Zoom calls forever without a break. So we scheduled for a break beginning to 2:45 to 3 p.m., a 15-minute break. So Aaron, do you have any announcements how people should join back?
No announcements. As noted, we were planning to return about 3 o'clock for the remaining presentations. I don't see any questions in the Q&A or the chat box. So if anybody has any for Dr. Desjarlais, feel free to enter those in now.
Yeah, just stay on connected. Like you can just be on mute and turn off your video so you don't have to use the link again to dial back in, because there might be some problems sometimes. So we'll see you all at 3 for the next talk. So I now introduce our next speaker. Is Dr. Julie Overbaugh. She's from University of Washington. She will be talking to us about profiling escape pathways for HIV antibodies. Julie?
Thank you. I want to talk a little bit about one project that we did under the Avant-Garde mechanism. Our overall interest in this Avant-Garde proposal was to try to understand how HIV evolution and variation impacts the function of the envelope protein and also its antigenicity. And I'm going to specifically talk today about the second part of that, which is understanding how changes in HIV envelope impact antigenicity with the goal of more effectively targeting envelope for vaccines and prevention of HIV infection. So I think most of you here know well that HIV envelope can evade antibodies through rapid mutation. It's a highly mutable virus. It also has other strategies for evading antibodies like altering conformation and glycosylation. And so this presents major challenges because HIV is such a genetically diverse virus, and it's hard to target the virus with antibodies. But of course, the good news is, and I'm trying to find my advanced slot, my advanced thing. There we go. The good news is that there are broadly neutralizing antibodies that can neutralize diverse viral strains, and they're elicited fairly regularly in infection, but not always. And everybody considers these to be a critical component of an effective HIV vaccine so that you can actually target the globally circulating variants. And also, these broadly neutralizing antibodies have been shown in the context of animal models to prevent infection and also to therapeutically suppress viral replication. So these are, for that reason, considered important in both prevention through vaccines and also potentially as therapeutics. But even though these are broadly neutralizing antibodies, they still are subject to escape. So HIV can escape these broadly neutralized antibodies, and if they do, this will compromise the efficacy that we hope to achieve with vaccines. And of course, we see this concept playing out with SARS-CoV-2 vaccines right now. So we wanted to understand the pathways of escape to try to develop approaches that might tip the balance in favor of the host over the virus. A lot of the traditional methods that have been used have been very useful in this regard. For example, people use structural studies where they look at complexes of the HIV envelope protein and an antibody, and they define how these two interact at the amino acid level. They define these contact residues, assuming that these are also going to be the amino acids in the envelope protein that could allow escape from the broadly neutralizing antibody. And then they use the structural studies to test this, and a number of mutations have been identified in this way. It's a very labor-intensive process and not complete. And what we also don't know or didn't know was whether these contacts that you capture in a structural study actually give you a very complete picture of escape pathways. The other approach people use is just to make individual mutations, either guided by structure or other biological reasoning, but this is very labor intensive and certainly not comprehensive. So we set out to develop a more comprehensive method to define escape pathways, and for this we used deep mutational scanning. And this work, I should say, was done in collaboration with Jesse Bloom, who had developed these methods in his lab at Fred Hutch for influenza virus. So deep mutational scanning is a method in which you mutate every possible amino acid in the protein of interest to every possible option. So that's just illustrated here with the wild type sequence shown on the top line. And you can see at the one position, we mutate every possible amino acid. At the next position, we do the same thing, et cetera. So you create a library that may be 10,000, 15,000 different sequences that capture every possible way the virus could evade and evolve. Okay, let's see if this will go forward again. Okay, so the method that we applied this to is called mutational antigenic profiling. This is a method developed by Adam Dingens, who was a joint graduate student in my lab and Jesse's lab. And Adam created this library of mutants in the HIV envelope protein. He generated this library in the context of replication competent virus. And then he put this virus library under antibody selection. So you have a library that is being challenged by an antibody. And as a control, you have that same library of viruses used to infect cells with no selection. You then look at the viruses that are successful in infecting the cells. And you see that those that were selected in the presence of antibody are those that were able to escape the antibody pressure. So you compare the sequence of the viruses that made it through this antibody bottleneck with the sequences of viruses that were not under selection. And you identify sequences that are enriched during this selection process. And that enrichment indicates that they are escape variants. So that's shown in the logo plot here, where you can see, for example, at one position, N671 in the amino acid, you see that a large number of viruses with a threonine at that position were able to be selected in the presence of antibody, indicating that that is an escape variant. And you can see by the logo plots that you can also not only see where escape occurs, but what amino acids are favored for escape. So you get a very granular view of escape pathways using this method. So the first thing Adam did was to apply this method to a well-studied HIV broadly neutralizing antibody, one that had been characterized structurally and with individual mutagenesis studies. And this is one called PGT151 that targets the fusion peptide and GP120, GP41 interface. And what Adam saw is shown in the graph at the bottom, which is that there were 3 regions where there's you can see indication of selection around 511, 611, and just beyond there in the envelope protein. So when you look at those positions in more detail and drill down to look at the escape pathways, you see that there are a number of individual amino acids within those regions that allow escape. And those are shown in the logo plots again, as I illustrated conceptually before. But I think what's particularly interesting about Adam's data is at the bottom of this logo plot, you can actually see the sites that have been identified in previous studies. These were extensive previous studies, structural studies, mutagenesis studies, high profile studies. And I think what you can appreciate as well, we captured all of those positions as being important in antibody interactions with PGT151 and escape. We also captured quite a number that were not previously identified. One example I'll just highlight is this position 648, where residues that change charge in the protein seem to allow escape based on this analysis. Of course, we wanted to know whether or not those mutations actually were causing escape in this proof of concept study. So we introduced the individual mutations back into the virus one by one, as is traditionally done. And we examined their ability to be neutralized by PGT151. And I'm just showing you here some data for that position 648. And what you can appreciate is that the 2 mutations that were most enriched in the deep mutational scanning study, the lysine and arginine at that position, were also the ones that most impacted neutralization compared to the wild type. And in fact, we looked at a number of mutations identified in this study and did the same thing across different amino acids. And what I'm showing you to the right in that graph is just the correlation between the results we generated using traditional neutralization with individual mutations in the virus compared to what we saw with enrichment in deep mutational scanning. And you can see there's a very high correlation between these 2 readouts. So with that success with a known antibody, Adam went on to look at a collection of antibodies that are from the 5 major classes of broadly neutralizing antibodies for HIV, the ones that target the CD4 binding site, the V3 glycan, the V2 apex, the fusion peptide, and MPER, as shown in the chart. And I point out here with the purple circle that a number of these are actually in clinical use or being tested, I should say, for clinical use. So these kind of analyses are not only important in understanding HIV replication and antibodies in tissue culture, but they really should apply to what we can understand from escape in a clinical setting. I'm not going to show you all the logo plots for these monoclonal antibodies, but I just want to show you a summary here and make a few points. So you can see the enrichment plots for each of these antibodies. And for example, the ones that focus on V3, PGT121, and 101074, we see enrichment at V3 in those antibodies. And indeed, for all the antibodies that we study, we do see the predicted epitopes using this method. But what's highlighted in the ribbon structures to the right of each antibody is, I think, an interesting finding from this work. And that is that if you look at the green highlights, those are the regions or the amino acids where we identified escape and where previous structural studies had shown this to be a contact residue. But what's highly interesting, I think, is looking at the blue amino acids. Those are ones where we identified escape, but where previous studies had not captured those just based on structural interactions. And there's a third interesting group, which is one where we see no evidence of escape, but where the structural studies suggest there's contact. So these kind of analyses, I think, illustrate the much more comprehensive nature of using this approach to map escape for broadly neutralizing antibodies. So in this method, what I've shown you is that this mutational antigenic profiling could identify known escape mutations as well as novel escape mutations not detected in structural studies. I want to just point out that this method is not just suited for studying antibody interactions with HIV envelope, but you can actually study other interactions as well. And we studied interactions of HIV envelope with CD4 receptor and also used this method to identify sites of what you could call escape or resistance from antiretroviral therapies. And we did a proof of concept study in this domain, focused, of course, on an antiretroviral that targets the envelope protein, because that's where we had made our library. And this is enfervatide, which is a salvage antiviral therapy that blocks fusion. And we did that because we had the ENV library, but I will make the point that this could be applied to other proteins as well, like polymerase, et cetera. So this just shows you the data from the selection that we did with the HIV deep mutational scanning library in the presence of this antiretroviral drug. And you can see that we again could clearly demonstrate resistance mutations or escape mutations using antiretroviral therapies, not just broadly neutralizing antibodies. And a number of these mutations, again, correlated with known mutations. And in addition, there were novel mutations identified using this approach. So what I hope I left you with from this first part of the study is the power of this approach called mutational antigenic profiling to provide complete mutation level mapping of viral escape. It massively increases the experimental throughput compared to structural studies in individual mutagenesis. And importantly, what we showed is it defines a functional epitope, which is different than the structural epitope. It can also aid in evaluating clinical trials, in refining vaccine regimens, and identifying drug resistance, as I showed you. But I will say there are some limitations to this approach. So even though it's high throughput compared to structural studies, it still requires large amounts of virus, and generally you can do one antibody at a time using this method. It also requires, as a readout, neutralization. So by definition, we're studying neutralizing antibodies in this experimental setup. But there's clearly increasing interest in non-neutralizing antibodies and antibodies that kill target cells through effector mechanisms. And those kind of antibody interactions would not be captured with the method as it was developed. So this really motivated us to think about how we could take this method and move it forward so that we could interrogate epitopes of both non-neutralizing as well as neutralizing antibodies. And I'll show you that in the next slides, and then also how we extended this method to study SARS-CoV-2. So the approach that we developed, we call phage DMS. And basically we take the same deep mutational scanning concept, and rather than putting all these mutations into the replicating virus, we put them into peptide sequences, introduce them into phage, and then we use the phage as to probe the antibodies, either monoclonal antibodies or antibodies in plasma, by looking for interactions through binding. So we capture those bound phage through immunoprecipitation. We can then deep sequence the immunoprecipitated phage and use those sequences computationally to first of all identify those enriched in the wild type sequence. In other words, those in the wild type sequence that bound to the antibody we were using, and that defines the epitope. And then within the epitope, we can compare the sequences with mutations to see if they were captured at the same level or whether they were reduced in binding, which would indicate escape. And again, we did a proof of concept study. This was a study led by Megan Garrett, a graduate student in my lab. And this was with a known, well-defined antibody targeting the V3 region of HIV envelope. And in the top graph, you see that in fact, what was enriched when we looked for wild type peptides that bound to this antibody in this format was exactly in the V3 region. And then when we looked at the mutations that disrupted binding, you could see evidence for escape or disruption of binding in what was considered the core motif of this epitope, GPGR. And that's shown in two ways in the graphs below. One is just sort of an inverse kind of logo plot on the left, and another is a heat map on the right. And the heat map, basically the red shows the amino acid positions and the specific amino acids that disrupt binding. Again, we wanted to make sure that what we measured in this assay would correlate with traditional assays. So Megan did tested individual peptides that were identified through the phage DMS in a competition ELISA and compared the binding disruption in that ELISA format with the extent of how these were selected in the phage DMS. And you can see there's a really strong correlation between these two measures. So just for the last couple slides, I want to mention that I think as many of us have done, taking what we've learned from HIV and applying to SARS-CoV-2 has been instrumental in really tackling this new pandemic. And it's really been a lot of what was done in the HIV field that has moved this needle forward so quickly. So we wanted to use phage DMS to profile the epitopes in the spike protein of SARS-CoV-2. So for that reason, we generated a phage library just like we had with HIV with deep mutational scanning. This library was almost 25,000 peptides. And we used that library to interrogate the responses in COVID-19 patients, in the convalescent sera from these patients, in a collaboration with Helen Chu, who leads a respiratory infection study called the Harvey Study Team, and in collaboration with computational collaborator Eric Matson's group, again, led by Megan Garrett, a student in the lab. This just shows you the enrichment of wild type peptides across different patients, showing you where they are on the structure in the linear sequence of the spike protein. So let me orient you a little bit here. The first thing is that on top, you can see the structure of the spike protein, the S1 protein and the S2 protein, much like GP120, GP41 of HIV. Two of the major epitopes that we identified as common across different patients were in the fusion peptide and in the linker region and HR2 region. And most of the individuals we studied had a response in this region of the spike protein in S2. In gray, you can see other regions where we identified epitopes that were more individual specific. And I will note we did not identify the major responses to the receptor binding domain, the RBD, because our method does not detect conformational and glycosylated protein responses. So I want to drill down. These are the responses that we saw for 18 of the individuals shown collectively. I want to drill down on the individuals and show you some interesting aspects of these responses. So these are 4 individuals now where I'm just showing you their individual enrichment plots on the left. And first of all, you can see that again, most but not all of them recognize the fusion peptide. The patient 9 did not show a response in this region. And almost all of them showed at least some evidence to a reaction to the HR2 protein, although patient 9 had a very strong response to that protein. On the right, I'm showing you the logo plots. And because here we're looking for a negative effect on binding, they're shown in the inverse orientation, but the concept is the same. And what I want you to appreciate is if you look at patient 1, patient 8, and patient 12, they all respond to the fusion peptide, but their escape pathways are all unique. And this is actually really exciting because it suggests that no one mutation that might be selected in nature would be able to escape in all different individuals. The responses are very different and very individual. The other aspect of this that I think is really interesting is where these epitopes lie on the protein. And I'm just showing you a structure here with highlights of the variable regions and the conserved regions. And what I think you can appreciate in the purple is that this S2 subunit where we map these dominant responses are in a more conserved region of the spike protein. And this is exciting because this may mean that this, if we target this more conserved and functionally constrained epitope, it could limit escape. And in fact, in the plot on the right, which I won't go into details on, we basically don't see evidence that the mutations that we've identified are becoming dominant in those circulating in nature. So that supports the idea that these epitopes might be remain more functionally constrained if they're under antibody pressure. So I'd like to conclude by just summarizing and hoping that you take away from this that this process of deep mutational scanning can be applied in a wide variety of ways to define escape pathways and viral resistance and other protein-protein interactions. It is comprehensive, and the results correlate well with other more traditional assays such as neutralization, ELISA, et cetera. I didn't show you all the data where we've applied this, but we've also applied this approach to a variety of settings, including being able to map and define the epitopes of new broadly neutralizing antibodies we isolated in the lab, and also in collaborative studies looking at vaccine responses to specific epitopes, particularly in non-human primate models. It's also provided insights beyond those afforded by structural studies, and it's much more high throughput, especially the phage deep mutational scanning. And of course, the work that we've done with COVID-19 infected individuals, we are extending to SARS-CoV-2 vaccine responses so that we can also anticipate any escape pathways outside the receptor binding domain that might be important in the vaccine responses. So I'd like to again acknowledge the people who contributed to this work. I think I've mentioned the key players. I'd like to again thank the NIDA AIDS research program and the AvantGuard for supporting some of this work and really getting us started in launching some of this deep mutational scanning work, which is, I must say, a very expensive experiment because of the deep sequencing. So this work was really quite essential to that. And again, thank you.
Thanks, Julie, for this wonderful talk. I'm going to ask you the first question. Do you have plan to looking at people who use drugs? We're just, getting sparse data that, people who use opioids may not mount immune responses to the same level as people who do not use drugs. Is there something that can be evaluated using the assay that you have?
And Kavi, are you thinking for the SARS-CoV-2 vaccine or infection
or even to future HIV vaccine candidates? So
yeah,
COVID would be good because we really want to understand, are people able to respond at the same level, whether it's pathogen or vaccine?
I think that would be very doable with this method. And I will say that, for the phage DMS method, we can, interrogate 100 samples at a time, which we couldn't do with the viral replication assay. So it is something where we could apply this to a population like that. If there were samples available, we could readily do that and would be interested.
That would be of interest to NIDA, certainly. Hi, Melanie.
Hi, this was a great talk, Julie. I just wanted to ask you a little bit about your general take on how the wonderful work that you and others are doing now with SARS-CoV-2 by going from previously acquired knowledge to SARS-CoV-2 and back. How will that affect the HIV vaccine in the future? And what is your prediction and in this? Because I think it's a unique opportunity and super exciting time.
Yeah, no, that's such a interesting philosophical question, really, Melanie. And I think, I feel like it's been such a good thing to take what we've learned and apply it to coronaviruses. I think obviously we cannot stop working on HIV, and those of us who do both now have to figure that out, right? And not completely crush our students in doing that. But I think looking at this, we're also learning a lot from COVID-19 and SARS-CoV-2 that's going to come back. So I think the circle will, in the end, benefit HIV, even though some of the energy for HIV probably got depleted a little bit this year, but I do believe that it's going to come back. I think the science, what we learn is going to be applicable. And obviously the mRNA vaccine, the fact that this concept is now clearly, so potent already tells us that probably that's where we have to spend a little more of our HIV time. And we didn't know that before. So we've learned a lot right there, I think.
All right.
Thank you.
Thank you.
Yeah, so it looks like we have an attendee with their hand raised. I'll go ahead and allow you to unmute Muthu.
Okay. Is there a question? Sorry.
Yeah, maybe no. Maybe he's not able to unmute. That's fine.
If you'd like to ask a question, Muthu, you should be able to unmute and speak.
So just to remind the audience, still can ask questions through the Q&A, and the panel members will get back to you with answers as much as they can before the symposium concludes. So with that, if there are no questions, I just want to make sure, Julie, that was a wonderful talk. Look forward to hearing more stories from this research in the future, and please do come back to NIDA. And if there's anything we could do to help support this research further to address more questions related to drug abuse, we would be really, really interested in engaging in those discussions with you.
Thank you.
So with that, I would like to introduce our next speaker. It's Dr. David Smith. He is from University of California, San Diego. He's going to talk to us about network-informed prevention. And I'm thinking HIV prevention.
Hello. Can you hear me and see my slides?
Can you switch it to the slide mode? Yeah, there you go. Perfect.
Yeah, can you see it and can you hear me?
Yeah. Good.
Perfect. Okay, so today I'm going to talk about networked informed prevention. I'm very humbled and honored to be in this group, and I really appreciate NIDA for the Avant-Garde Award. It has changed my career. My name is Davey Smith. My pronouns are he, his, and him, and I'm at UC San Diego. I'm going to talk a little bit about evolution because everything makes a lot more sense once we understand it through the lens of evolution. And when Darwin established that all species of life were descended over time for ancestors, that evolution resulted from a process of natural selection, then this change can be observed in the genetic code. And my project was about understanding how to use that to our advantage. And first off, starting with HIV among all the RNA viruses, There's low fidelity and then there's high fidelity viruses, RNA and DNA, and HIV has a pretty good mutational rate. Julie talked a little bit about this earlier. Hepatitis C has more, but there's a spectrum, but there's enough there for perhaps we can use it to our advantage. And one of the things we did, first off, looking at a Chinese study, we saw that there was differences in mutations in different risk groups. And here is the percentage of viruses that we found in different populations. And some of these clusters had different mutations with lysines and et cetera in different groups. And we could saw that people who used injectable drugs had E9296 strains, while MSM in this population had only K92 and E96 strains. So this gave us some interest in could we track what happens over time within these individuals in terms of mutations. So to step back in terms of evolution, perhaps we could use evolution to our advantage here. And evolutionary processes leave measurable footprints in the viral gene. And the genetic distance between two viral gene sequences reflect their relatedness. So you could look here at envelope or GAG or PAL for HIV, and you could see that these trees look quite different and spread out depending on their genetic relationships amongst each other. And each of these represents the different clades. So viruses within clade C look closer together than viruses that are C to A or just within perhaps A. For example, I'm genetically closer related to my mother than I am to your mother. And if we were to do sequencing, we could figure it out. But in HIV, that difference is on steroids, and we can really sort this out in a phylogenetic way. And we can make maps of this transmission network, how HIV has gone from one person to another person to another person. And this is really good work from Sergei Kosogasi-Pon and Joel Wertheim, looking at how to make an HIV transmission cluster engine, turn phylogenetics into an ability to look for clusters. So each person has their own unique virus that's adapted to them. We can then use phylogenetic or genetic distance methods to sort it out who might be closely related within a transmission chain to come up with clusters. And then we can use that information to, let's say, investigate outbreaks. So here we took 288 HIV sequences that were generated between 2004 to 2016. About half of them were men who had sex with men, a third of them were women. 40% reported transactional sex, and 28% reported injection drug use as their risk factors. And 13% of these people were recent infections. And then we analyzed these sequences using distance-based methods to figure out that 37 transmission clusters were within this group, involving 122 sequences. And then while we were working on this, we saw in part of our avant-garde work, we saw a great increase in the number of new infections in mid-2016. So we're like, okay, what does that look like? Well, since we could map it out, and we can map it out by risk factors, we started making these little clusters. So this group right here had very closely related sequences. This group here had closely related sequences, and each of these little connections we could make based on their genetic distance between their viruses that were sequenced at a certain time. And what we noticed is that there were big groupings of, here's female sex workers, and in the green were heterosexual, the blue were men who had sex with men, and the light blue were bisexual. And then people who injected drugs were in the red big squares. We started to see a pattern that there was an increase of people in hotspots who were using injectable drugs. So what was going on? Well, this study was conducted in the border region between San Diego and Tijuana. And specifically, we had a population of people who injected drugs in the Tijuana River Canal, El Bordo. And this is a picture of what it looked like before. And then this is what happened in late 2015. They went in and they cleaned it up. We're like, okay, what was going on here? This was associated with a time when we actually saw an increase in the number of infections in that cohort. What we saw was that unfortunately, after moving from the canal region, the people who injected drug found new galleries, which were safe from the police. So the police were the ones that came in and cleared out those galleries. So they just moved out into the suburbs, basically, and found new galleries to shoot. But this was also away from the NGOs that were giving harm reduction materials in terms of basically needle exchange at that time. And this, we're pretty sure, increased the risk of infection that we were seeing within those galleries. So that was one really good way of figuring out that we could see when there was an issue. Another one is, can we intervene on the HIV transmission network once we kind of have it mapped out? And one things we notice when we map out these clusters for transmission networks is that people have different positions within the cluster that we identify. Some people are connected basically to everyone else in the cluster. And then some people are only connected to some people in the cluster, or maybe even only one person in the cluster. And we call this degrees. So if this person is connected to 7 other people who have infection, then that's 7 degrees, versus a person who's only connected to one person who is infected, that's a 1 degree versus 3 degrees. So now we have a simple but qualitative measure where we can see what the centrality in the network might make a difference. So our hypothesis was that people who were more central in the network, i.e. had more degrees when we mapped it out, were more important to intervene on for prevention. And we did a nice study in a China MSM acute infection cohort that had been collected for a long period of time and had been tested on a regular basis. And all the participants had CD4 counts and HIV viral loads and genotypes that allowed us to look at it. And our question was, well, what if we targeted antiretroviral therapy? This was at a time in China where they didn't give everyone therapy based on CD4 count and viral load. This was all done retrospectively, but we wanted to see whether or not degrees made a difference. And our strategy was, if you just treated all the participants, how many people would you have to treat? How many cases would you have prevented? And then what kind of prevention efficiency would you see? So then we wanted to say, well, okay, let's see if you targeted the people who had the really low T cell counts. That would have been 9 people, and we would have prevented 1 case. And really, any of the CD4 counts didn't give us much prevention efficiency. In fact, it was less than if we just treated everybody. If you look at viral load, which is definitely a predictor of transmission, if you gave it to people who only had really high viral loads, 10 to the 5th, you would have treated 36 people, but only would have saved 3 people within our transmission network an infection, and that was 8%. Versus greater than 50, it would have been 57 people you would have treated and got to 23%. All of these are less than if you just would have treated everybody. So there was no benefit for using these other measures as surrogates for prevention targeting. So then we go back, okay, well, do degrees matter? So does the network tell us anything of who might be important for targeting prevention efforts, such as increasing adherence to antiretroviral therapy and other things? And I'll talk about those in a second. So in this group, we have the number of participants on this y-axis. We have the efficiency on this axis. We have the number of degrees of a participant in 2009 and how many infections would have prevented in 2010. If you treated everybody at 120 people, you would have saved this many number of infections. But if you started treating everybody who had 1 degree or more, you would have had this efficiency. And as you can see, that there's basically a good stepwise progression, that the more degrees that somebody had in the network, then you would have prevented more infections in the future, with a 55% prevention efficiency of targeted antiretroviral therapy for those who had the highest degrees. So even though there was only few people, I think 15 people who had these high number degrees, if you could get them on therapy in this setting or some other prevention, then you would have the biggest impact in future transmissions within that network. So then we came up with an idea, and we basically structured this to where we take HIV pulse sequences, which are used in standard clinical practice to look for transmitted drug resistance before somebody goes on therapy. And then we put that into a real time in the background of all sequences. And then this gives us a network for our local community, and we can start to map out what degrees everyone has in real time in this network. We then generate what we call a transmission network score. If the score is high, which means they have a lot of degrees, or if it's low, we just follow them. But if it's high, we generate a risk profile in an intervention plan. And that's where we look at the demographics, the risk behaviors, the venues to where they're might be at risk. We look at the estimated duration of infection, acute, early, chronic, and then we also add in CD4 and viral load, since we have them. And then we propose a ring intervention where an intervention team of a nurse, social worker, test counselor, whatever that prevention intervention looks like, and then target venues such as HIV testing, counseling, STI testing, and needle exchange. Another big point about this is these methods also allow us to know whether or not we can be successful, is whether or not our intervention is actually successful, looking at the phylogenetic lineages that die out over time. So it's a real iterative process. You can say, okay, if I target, let's say, bathhouses or shooting galleries more than I target something else, I can see those lineages die faster, i.e. less transmissions going forward than I would expect, or not. And this data, I'm proud to say, is part of the RESPOND pillar for ending the HIV epidemic that the CDC now has adopted a few years ago. And this RESPOND is to respond quickly to potential HIV outbreaks to get needed prevention and treatment services to people who need them. And through this avant-garde, we worked for multiple different groups, including the CDC for establishing the pillar. And Joel Wertheim, who got a K Award with me as his mentor along these lines, is really ingrained in this and has done amazing work. We also worked with the China CDC for MSM and substance use. We've worked with India for their NACO for substance use and controlling HIV outbreaks. The Mexico Center for Infectious Disease Research is ongoing right now with big collaborations on establishing their response for molecular epi. South Korea CDC has similar plans, as does the Mozambique Ministry of Health and Cape Town HIV control program. All are up and running. So another one, since we are in the time of COVID, we've adapted a lot of the methods and stuff that we learned early on in sequence analysis and molecular epidemiology and brought in new techniques. And one of those big techniques is called phylogeography. And here I'm just going to talk a little bit about determining the dynamics of the SARS-CoV-2 across the border. I talked about HIV across the border earlier. Can we do the same thing with SARS-CoV-2, which actually has a higher fidelity than HIV for its mutational rate? And then we were very interested in the impact of human mobility on the viral dynamics that we saw. And here we looked at between January of last year to January of this year, we had almost 12,000 SARS-CoV-2 complete genome sequences downloaded from this public database that has been absolutely amazing. It's a worldwide effort to get these sequences out into the public domain so that researchers like myself and Antoine Chalone, who did a lot of this work, can access them. And here we were interested in California and Mexico. And you can see that there is a big lopsided amount of sequences that came from both. But the nice thing is we have developed techniques and ways to account for that. But in these sequences, we combine them with a representative set of non-California and Mexico sequences. So it's a very large database, and we identified clades, i.e. basically you can think of clusters with only mixtures of California and Mexico sequences. And then we did another analysis to come up with time to most recent common ancestor lineages for each of those clades that had 2 or more sequences that were attached to it. And we came up with a date of when that occurred. And then we looked at clades that were greater than or equal to 3 sequences originating from 2 different locations. So if there were 3 sequences in San Diego, that didn't do me any good. But if I had 2 sequences in San Diego and 1 in Baja California or 1 in Los Angeles, then I could include that on my further analysis. Decreasing the amount of data that we have to analyze that's informative greatly gives us a chance to actually get it completed, since these are large computationally intensive data sets. But then we went on to do phylogeographic inference for these clades using BEAST, so a Bayesian analysis, sequence analysis. So here we had 96 clades of greater than or equal to 3 sequences, including both California and Mexico that were identified. The ranges were from 3 to 48 of cluster size. And then we did a discrete phylogeographic analysis looking at the viral migration network across the border. And we also looked at the migration pattern. So we knew how many, in general terms, how much human migration was going back and forth from across the border. And basically what we found, we could draw these, what I call these intercontinental ballistic missiles, but we can see which of these places were connected. So we had a few sequences in San Diego, and we have a few sequences in Mexico City, and then we would draw a line. Or if we had some in San Diego and Los Angeles, we would draw a line. And these are the transitions that we saw. And we can add, since we're using Bayesian analysis, we can use what we call Bayes factor analysis to come up with statistics, which of these are actually supported transitions, which has greatly increased our ability to have confidence in what connections that we actually see. And basically what we saw was that most of the transmissions that we observed were coming from Baja California, which is in Mexico, over into San Diego, and a little bit above into San Francisco or to Ventura, but also within Baja to other parts of Mexico we could also see. What's interesting is there were tons of flights that went back and forth, but the land border seemed to be really important for a lot of the observations that we saw. And it didn't really matter how many sequences we had from each of these. It was all about the clades and the connections between those two. Because there were a lot more sequences that were used in San Francisco versus San Diego, but still the migration was mostly from Baja California to San Diego. So that's SARS-CoV-2, and that work is directly related to all the great stuff that we did within Avantgarde. But another component of this is along the same lines. So you can think of big things happening between places as phylogeography, but there's also little things that happen within a person that has HIV. And this is back to HIV. And this is a project that we have here in San Diego that's part of our last gift cohort. So these are people who are terminally ill, who they have HIV and they're terminally ill of something else, such as cancer or ALS or cardiovascular disease. And we follow them pre-mortem, and then when they pass away, we do a rapid autopsy and collect specimens all across their body. So we get blood before they die, we get lymph nodes, all the gut, etc. you can see. And then each of those tissues, we can sequence it. And since we followed them ante-mortem, we have blood beforehand, and we can also get genital secretions and rectal biopsies, etc. And some of these people actually stop their therapy. They're passing away and they don't want to take their therapy anymore. So they sometimes stop their therapy. So we'll actually be able to observe rebound as well. But independently, we can sequence all the HIV from all these tissues, and then we can use very similar techniques to figure out, what are tissues are most closely genetically related within a tissue, within the same person, to see where HIV might migrate between all these different places. So it's a different ICBM map within a person. And then we can actually look at genetic flow with a statistical support called a Bayes factor. And here you might see that within the brain, it goes to one other places, maybe the prostate or the rectum is where we see it next, or the big flow almost always is PBMC to lymph nodes. And we can drill down into very specific things, looking at, let's say, the blood-brain barrier. How does the basal ganglia track to the rest of the basal ganglia, or the blood clot going to the hippocampus, or within the esophagus going to other parts of the gut, which is very interesting. And then the genital tract is another barrier. What are the ones that are connected? And as you can see, there's a lot less connections in the genital tract. And now we're doing experiments underway to evaluate this same HIV migration in relation to people who take opioid. During the last parts of people's life, they're often under considerable pain, and there's lots of opioids that are being used in that setting. And that gives us an opportunity to look at those concentrations in relation to what we might see when somebody stops their therapy, or even when they continue their therapy, to look at migration. We do see quite a bit of provirus that looks like to be moved within the blood and then seeding within tissues, even though someone is completely suppressed with antiretroviral therapy. So I just want to acknowledge my collaborators in South Korea, in China, in India, in South Africa, who really adopted a lot of these techniques, and we publish quite often with them, but really worked closely with their ministries of health to get their molecular epidemiology programs together. And a lot of that work with Joel Wertheim and getting his HIV trace out into the world has really greatly increased its utility. And in here, developing new methods along with Sergey and Sarah and Antoine. And of course, Susan Little, with a lot of big programs here in San Diego, still going on with our health departments on trying to really use these tools for the maximal prevention efforts as possible. And then I'll stop there and take questions.
Thank you very much. Excellent presentation. We have some questions coming in the Q&A. So, Davey, also, we are very proud that we've been able to award a DP2 recently to another one of your team members who is working on the last gift cohort.
Yeah, we're really thankful for that. Sarah's going to do great work with that DP2.
We're looking forward to her progress as well. So here's a question. So you mentioned treating individuals with high degree centrality. Did you look at this by any other centrality measures, such as betweenness or eigenvector centrality? Yeah. And what are your thoughts about, there's a second component to the question, what are your thoughts about treating all individuals at popular injection venues, and whether that might further impact network level prevention?
Yeah, so first off, I think I am for treatment of everybody as quickly and as soon as possible that someone is diagnosed, to get them therapy as quickly as possible. That's first of all. And then the second one, we looked at a whole bunch of measures, and we still are looking at betweenness and Eisensteiner and various centrality metrics. It turns out that just degrees is the simplest and probably works just as well. However, I have to say that there is some issues around homophily with different groups, such as you can think of the classic one is a man who has sex with men and with women, and that difference bridging effect seems to be very important for prevention efforts. And in every single model that we do, it definitely comes up, but in real world practice, we see a signal there, but it's not as strong. And I think that the reason for that is we're not as good at identifying everybody in the network that might be affected. But the other thing about I used antiretroviral therapy as the intervention, but there's a whole bunch of things such as needle exchange or other harm reduction services. It was just cleaner in that Chinese cohort to use antiretroviral therapy, but we're working with lots of people here in San Diego and other harm reduction techniques intervening on the network.
Maybe perhaps linking them to medications for opioid use disorder. So there is a question from Sandy Springer. She says, did you look at adding PrEP availability in prevention model as well as MAUD for those with OUD in prevention model, including those without HIV?
Yeah, so PrEP is,
Yeah.
yeah, so PrEP is really, really good. So in the network that you see using molecular epi, you're only looking at people who have the virus. But with Susan Little's work and with Joel Werthein's work, they're looking at people who are connected to the network but are not infected, and you can come up with other degrees of connectedness along those lines. So looking at risk networks, and then where does PrEP attach to those people who are uninfected but perhaps exposed to your high-risk network. And that work is fascinating and is underway.
Great. There's another question here about which portion of the basal ganglia that you talked about.
I could probably spend way too much time talking about different parts of the brain, but we had multiple different pieces of the basal ganglia. We did not find a particular part of the basal ganglia that made the biggest difference. It is quite variable. It is the basal ganglia HIV concentrations is quite variable across people.
I think that's pretty much everything. Just make sure I'm not missing anything that came through chat. Well, congratulations, Davey. Excellent work, and keep in touch.
Thank you.
We look forward to hearing more from your group in future.
Yeah. Thank you.
So that leaves us to the last presenter. I'm really waiting for her talk. She has some cool story that she is going to present or talk to us about. This is Dr. Melanie Ott. She is from University of California, San Francisco. She's going to talk to us about HIV and aging from molecules to tissues.
Well, hello, everybody. Let me just make this big. Do you see everything?
Yes, it's good.
Perfect. Thank you, Kavi. Thank you very much for the invitation and the kind introduction. Thank you, everybody, for staying until the last talk. I'm a virologist from San Francisco. And I really want to thank NIDA for the Avant-Garde Award, because it has been transformational in two ways. First, it has really helped us to trust, to really focus on the molecular biology of aging and seeing how HIV and drugs of abuse would interfere with this. And then secondly, it has also allowed us not only to focus on the immune system, but eventually to branch out into tissues. And I will show you more about this during the talk here. So just to step back at the beginning to sort of review our amazing progress that we have done over the last 2,000 years in terms of enhancing our lifespan and also our health span, but particularly in the last century, where we made a huge jump in terms of life expectancy, and that is mainly due to water cleanliness and vaccination. However, we know that one group has been left out from this, or has actually regressed in this enormous progress that we have done, and these are people living with HIV. And we all know that this is not because of AIDS. This is just by people who are living chronically with the virus are experiencing an accelerated rate of certain diseases of aging that basically curtails their life expectancy. And it has been estimated that it's at least 5 years of lifespan that is actually taken from people living with HIV. And this is increased even further if it's HIV positive drug users who are on average losing up to 10 to more than 10 to 15 years of their lifespan. So our goal was to really understand what is this, what are the mechanisms of this decrease in life expectancy, and how could we find ways to target these molecular mechanisms to potentially reverse it. And as I said, we wanted to focus on the molecular biology of aging, because we know from a really burst of research activity in the past 10 to 20 years, that there is a program in us and in our cells and every organism that controls aging and lifespan. And there have been many molecules and proteins being identified that could potentially that play a role in this process and could be potentially targeted therapeutically. One of those are the so-called sirtuins. These are a group of protein deacetylases that are highly conserved between bacteria and mammals, and specifically in yeast and C. elegans have shown that they increase lifespan if you increase the copy number of the SIRT2 protein. Now, there are 7 homologs of this SIRT2 protein in humans or mammals. The first one, SIRT1, is the best studied and probably the closest in function to the SIRT2 protein. It is, as I said, a deacetylase, and it's a very unique deacetylase because it's directly connected to the metabolism of the cell by consuming NAD for the deacetylation process. And it has many downstream targets that link it to many biological processes. We have worked on this protein extensively and have sort of contributed multiple unique aspects to its function. One of them is that we found that it actually directly deacetylates the HIV TUT protein and regulates its function, but we also have worked extensively on its role in NF-κB and in inflammation and the connection with HIV infection. During this talk, I want to draw your attention to the FOXO proteins, which are very important SIRT1 downstream targets that we have discovered through the course of our Avant-Garde Effect Award to really play an important effect in the biology of SIRT1 in the immune system. And this is very satisfying because the FOXO proteins are really the closest linked to human aging, and the way that GWAS studies have shown that variants in the sirtuins or in SIRT1 are connected to are found in higher incidences in centenarians. Now, for us, what was really striking was that we found that TUT interacted with SIRT1. It was also a target of SIRT1, but it also had a very unique inhibitory function on the SIRT1 deacetylase activity because it binds directly into the deacetylase domain of SIRT1 and is here in a biochemical assay a very potent inhibitor of its deacetylase activity. In fact, it's even more potent than its natural inhibitor, nicotinamide, in the cell. And it was very striking to pharmacologists when we showed this to them at the time. This in the cell translates now in increased acetylation of some of these downstream targets, and acetylation is usually associated with increased function. And so when we look at NF-κB P65 here, which is acetylated at lysine 310, we can mimic this by overexpressing the acetyltransferase P300. You see nice acetylation if you look with an acetylation-specific antibodies. If you overexpress SIRT1, this is decreased. But then if you co-express increasing amounts of TUT, you see that this completely neutralizes the deacetylase activity of SIRT1, really showing the potency of the TUT effect in these cellular and in vitro assays. So we have since then focused a lot on this so-called TUT-SIRT1 tango, that's how Mattia Peterlin called it at one point, and have published many papers showing that that it is really relevant for the biology of immune aging, but also HIV infection. And I want to give you some vignettes of this work in this talk. So we have focused, as I said, primarily on the immune system and the fact that our immune system ages over time, and we know from many studies that this is accelerated with HIV and, for example, methamphetamine use that transforms actually the immune system of a young person more into the phenotype of an old person. And this is mainly shown here schematically by trying to illustrate that what we lose over time is, of course, our pool of naive T cells, because we lose our thymus, but we also lose our repertoire in terms of the memory T cells that we have, because they become more and more focused on very specific clones, or they're very much determined by sort of clonal expansion of certain memory T cells that over time become more and more terminally differentiated. That means they do not respond anymore properly to antigen exposure, and they instead have more of an innate cytotoxic phenotype. Now, when we wanted to see whether this HIV, this SIRT1 connection is in any way relevant outside of an HIV-infected cell, we looked now in general into the distribution of these different memory T cell phenotypes, the naive, the young ones, the CD28 positive ones, which is the marker that are used to differentiate them, or the CD28 negative ones, which are the ones that are terminally differentiated. And just by looking very naively at whether SIRT1 is in these cells, we found that actually SIRT1 is very lowly expressed in these terminally differentiated T cells. There's actually a gradient of expression. It's highest expressed in the naive ones, then it goes down in the memory T cells, but then the terminally differentiated ones, they have very low levels. And this is shown here in the in this analysis of protein levels. And we have recently solved the mechanism, what is going on here, together with Shelley Berger, who is a close colleague at UPenn, and who has looked at SIRT1 decrease over time in different tissues and has elucidated the mechanism here, and finds that it's mainly degraded by macroautophagy. So in terms of over time, autophagy somehow increasing, and SIRT1 being basically degraded. But if we block macroautophagy here with a specific inhibitor in the CD28 negative cells, we can recover the almost normal levels of SIRT1 in these cells. Now, when we looked in CD28 cells now downstream and wanted to see what is the effect, what are the downstream targets that are mostly affected by the loss of SIRT1 in these cells, we found that FOXO1 was really a very obvious target, because you can see very easily it has exactly the same distribution as the SIRT1 protein. It is basically also lost in these cells. And it's very easy to understand, because SIRT1 usually deacetylates FOXO1 in the nucleus, and that keeps FOXO1 in the nucleus. However, when SIRT1 is gone, FOXO gets exported, is hyperacetylated, it gets phosphorylated, and then it is basically in the cytoplasm degraded by the proteasome. So to just make sure that this is really the way how this is working here, we downregulated, or we edited SIRT1 with CRISPR in primary T cells here, and that led immediately to the loss of FOXO1 as we had predicted, really establishing that this is sort of the course of events that is taking place. And in contrast, when we use CD28 negative T cells and overexpress SIRT1, here we nucleoaffected a recombinant protein into these cells to avoid some cytotoxicity. We could recover and stabilize FOXO1 in these cells, really showing that there's a SIRT1 FOXO1 axis that is very very prominent in determining biology in T cells. Now, I spare you all of the detailed experiment, or most of the detailed experiments here, because this is published. But what we found is that a very important function of this 31-FOXO axis is that FOXO suppresses the glycolytic capacity of these cells. So and thus also suppresses cytotoxicity in the resting state of these cells. So that is really what we believe is the break in a resting T cell that is basically preventing these cells from becoming glycolytic, but instead using oxidative phosphorylation. However, we know over time, or as I showed you in HIV through inactivation, and we believe also through through drug use, you actually accelerate the loss of SIRT1 in these cells. And that leads to terminal differentiation in the way that it also leads to a loss of FOXO1. It unleashes the glycolytic capacity of these cells, and these cells become very strongly innately cytotoxic. So we have shown in that paper here that we can actually manipulate that axis here in the way that we can go into CD28 negative cells. We treat them with resveratrol, which is an enhancer of SIRT1 function, and that somehow partially restores the activity. We can do this also with NAD mimetics when we replenish or enhance NAD concentrations, and that also increases SIRT1 activity, and that also really puts back the brake onto this glycolytic activity and the cytotoxicity of these cells. But one of the things that we also discovered was that there is a very, very specific inhibitor of FOXO1 that actually mimics this accelerated course of action that I'm showing you here, blocks FOXO1, and that thus unleashes this glycolytic capacity and cytotoxicity. It's a very specific inhibitor. We were very worried that it would cross-react with other FOXOs. However, in this cellular thermal shift assay, you can see that only FOXO1 is reacting to the drug. And it's a very potent inhibitor, because we can immediately, just by the addition of the drug to primary T cells that are resting, induce these glycolytic activity, or unleash this glycolytic capacity here, shown by this seahorse analysis, where when you treat the cells with a mitochondrial poison, you increase basically glycolysis, while under untreated conditions, the cell basically crashes. And at the same time, you can see that this also leads to a significant increase in granzyme B production, transcription and production, indicating that indeed the cytotoxicity of these cells under resting conditions is greatly enhanced. So we have looked at this and now made back the loop to HIV, because we wanted to know, is this only something that we observed in CD8 cells? Because CD8 cells were the easiest to analyze. We basically do all these experiments in human CD28 positive or negative cells, because mouse and human immune aging is significantly different. And so the question now was, is this also applying to CD4 cells? And I can tell you, we see exactly the same thing in CD4 cells. We just have less quantities in cells that we can recover from PBMCs. And we also wanted to see, what is the effect now on HIV if we manipulate this axis? Is this axis in any way benefiting or not HIV infection in these cells, and specifically latency? So we have just simply what we did is we used a cell line, a JLAT cell line that was established here at the Gladstone Institute, that gives us a very easy readout. These cells have basically a latent provirus with GFP in them, and then you can activate it. It's a deficient provirus. You can activate and see whether transcription is activated just by looking at GFP. And you can see if we give increasing amounts of the FOXO1 inhibitor, and after 24 hours, we don't see anything, but after 48 and 72 hours, we see a very, very strong activation, reactivation of the provirus in these cells, even better than TNF-alpha, which we usually use as an activator. And we have shown that This is done in many different cell lines, and I'll show you in a minute also in primary cells, but there's also a very strong synergy actually between TNF-alpha and the FOXO1 inhibitor, where you see that, that small amounts of both actually synergize incredibly well, indicating that they might actually work through different pathways in the cell. And I'll come back to this in a minute. Just here to show you that also ex vivo infected primary CD4 T cells respond to this. And in patient cells, we can also see significant reactivation. However, we see again the best reactivation when we give small doses of a PKC agonist here in order to pre-activate the cells together with the FOXO inhibitor. Now, to cut a long story short, we had thought that this is all metabolically driven and that there's a direct connection between HIV and the glycolytic capacity that I showed you of these cells. However, it was very, very striking that this was not the case. It was actually all going through a different pathway, and all, especially after we did RNA-seq, everything pointed into the direction of ER stress as the mediator of the reactivation of the virus. We could see that the FOXO inhibitor strongly activated ATF4, which is one of the proteins that during ER stress is still translated and regulates downstream ER stress countermeasures. And we could see that, and I'm not showing you the data here, but lots of studies have shown that it's actually going exclusively through the PERK arm of the ER stress pathway, not through other viral infection or amino acid deprivation stimuli here. So this was very surprising to us because we had really thought completely in a different direction, but this is what the data showed us. And we found that ATF1 directly can bind to the provirus in CHIP assays here. This is something that has been previously reported, and it was completely in the absence of NF-kappa B activation. But NF-kappa B activation was intact if we treated with TNF-alpha, but it was in the presence of a very strong NFAT activation. So these are two very strong drivers of HIV transcription as transcription factors. ATF4 is something that has not been known to be a primary driver. However, these are very important regulators, and it was very striking to us that there was an either no NF-kappa B, but a very strong NFAT activation here. So in summary, I think what we found in the study is that this SIRT1 FOXO axis here is very important to diminish the ER stress also in cells. And that leads basically to no activation of the PERK and ATF4 pathway or calcium released from the ER and NFAT activation. So HIV transcription is kept low and latency is big. However, if we remove this axis again, either through natural or here a therapeutic intervention, we see that this unleashes ER stress, and that leads to activation of PERK, production of ATF4, and direct activation of HIV transcription through both ATF4 and NFAT, and basically latency is overcome in these cells. So one of the things that we were really excited about, and that I just want to point out here, is that the connection here between FOXO and ER stress lies in the regulation of autophagy. So there is basically a feedback mechanism potentially to SIRT1 production and stability, but there's also a very strong phenotype here where when we look at direct protein aggregates in the ER of these resting T cells, we find that they do have these aggregates. But when we take FOXO away here with the inhibitor, or if we knock it down, we see that these aggregates accumulate, and we think that induces the ER stress and is a very strong, so far understudied pathway that regulates transcriptional activity and biology in these cells. We think that ER stress, calcium, is really important, mimicked here with the FOXO inhibitor, is really important to transition from a quiescent cell into which harbors a latent HIV into a cell that is basically reactivated without activating the cell per se. But we're really interested in this role of the ER stress or the UPR and the connection with SIRT1 in the question how this is a new player in the immune aging biology, in the fact that we predict that in memory T cells, in young memory T cells, the ER stress is very low, but in the CD28 terminally differentiated T cells, ER stress is unleashed because SIRT1 and FOXO1 is gone. And we are very interested in seeing how modulators of ER stress or this axis that I have shown you above could now in any way manipulate this pathway and potentially rejuvenate some of these terminally differentiated cells. We're very interested in the role of methamphetamine in this process. There's many studies that have linked this drug with ER stress. And we also have started a new collaboration with Faris Papa here at UCSF. He's an ER stress expert which has started a very interesting drug development program by trying to really surgically interfere into the different arms of the ER stress to see what it does. So we're very excited to combine these two new aspects of this biology to see how we can move potentially revert damage that is caused by drug use, HIV, or natural age on these T cells. But I'm telling you, we have been sort of focusing on these T cells painfully and in the past few years to do all our studies. But aging is not only in the immune system. We think that it's an important driver, but we also think that it is in many other tissues. And so what the Avantgarde Award has allowed us to do is to establish the tools in the lab to really look into these different tissues. And we have heavily invested into different human organoid systems that allows us now out of stem cells to reconstruct many organs in the dish. We do this for the lung, the liver, and the gut in the lab. But we also collaborate with our iPSC experts here at Gladstone for other, for example, neuronal progenitor cells, or we collaborate with other colleagues here to use these organoids now for infections, but also very importantly for co-cultures with HIV-infected T cells. And we think that this is a good way to directly test what effect drugs of abuse, for example, have on direct tissues.
Dr. Ott, just a few minutes before the Q&A.
Okay, perfect. I'm almost at the end. I just wanted to say that this has allowed us to be potentially significantly contributed to SARS-CoV-2 also in the last year, because we have used many of our models, lung, gut, and heart, to be infected. Here in green, you see SARS-CoV-2 replication centers, but we have particularly focused on co-infections of the liver, hepatitis C infected liver, and HIV. Just to say that, of course, hepatitis C is a very important aspect, especially for intravenous drug users, because there's the very high infection rate, co-infection rate. And I also have to say that I have waited 20 years for the moment now that I can actually study HCV and HIV, two viruses that I have a long-term investment in on the molecular side, that I have a way to really meaningfully study this in the dish. So we have developed organoids from HCV positive and negative donors. These are liver organoids that I'm showing you here, either in the proliferative or in the differentiated state. There's not much of a difference, but the interesting part is that we saw that some of these organoids were actually retaining the virus in the tissues when we built them in the dish. So we had a chance to actually start to study HCV infection in these different organoids, while others who came from HCV positive patients did not have this, did not carry forward the virus. However, we could use virus from the same donor and infect these cells very effectively in vitro, both in the stem cell and the hepatocyte-like state. We did single cell RNA-seq. I might skip over this. So very interesting changes in terms of differentiation, but we're mostly interested in finding out whether these cells actually are antigen presenting cells by looking at HLA expression here and also co-culturing them with non-HLA matched T cells and seeing that that immediately induced proliferation and activation of these T cells. But eventually we used HCV-specific peptides to pulse these liver organoids and then co-culture them with a peptide-specific clone. We used a specific microfluidics chips that hosts the organoids in the middle, and the T cells here, cytotracker green labeled in the outside channels. And you see that these are then migrating into the channel to surround the organoids. And we could see that there's, great coexistence of T cells and organoids in the absence of peptide. However, when we pulsed it with the peptide, you could see that there was immediate cytotoxicity and destruction of these organoids that really validated this system as a good system to study. Now, what is the effect of an infected liver on the generation of on immune aging and the generation of these CD28 negative T cells, but also vice versa, what is the effect of an infected CD4 positive T cell on the HCV positive liver, and the same thing for HIV if we infect these cells. So just my last slide, just to summarize, I think it was a very interesting and an exciting journey in the past 7 years now. Where we were really focusing exclusively on the question how molecular pathways of aging would contribute to the reduced lifespan of HIV-infected individuals or in HIV-infected drug users. And I think we have identified a series of targets that we're following up in terms of what would be a good reversal strategy to prevent some of these diseases of aging that are occurring at accelerated rate in HIV-infected drug users. So with this, I want to stop. I had a great group of people in the lab over the years who have worked on that and carried the baton with great collaborations with colleagues here at the Gladstone in the Bay Area, with clinical colleagues who gave us the liver tissues to start the organoids, with Eric Verdin at the Buck Institute as a preeminent aging institution, but also with the Jen Zuckerberg Hub here in San Francisco. And again, many thanks to NIDA for the generous support, because without this, none of this would have been possible. Thank you very much.
Excellent talk, Melanie. Thank you. I'm going to ask you a real quick question and then go through the questions that are coming in Q&A. Have you looked at elite controllers or people who have long-term, have been under odd and strong virus suppression? How does the T1 or FOXO activity look like?
That's a very good question. So what we have done is we did look at them, and we looked at different HIV, groups of people. What we find is that if we look at their CD28 negative cells, we see exactly the same. We see that they all lose basically FOXO and SIRT1. There's no difference qualitatively in these cells. However, what we're currently doing is we're looking in the precursors of these cells, all the different, immune subtypes, and many of the, much of the research that was also presented today is extremely interesting because there's still a great need for better granularity to see how these cells actually arise and what are the precursors. So we are predicting that we see changes either in the numbers or in the quality of SIRT1 and FOXO1 expression in these precursor cells, and that leads to basically that end stage, which is the same for all of them.
That's great. Would love to know the answer to that. And you are going to love this question. Does TAT work better as acetylated or unacetylated in inhibiting SIRT1?
That's a very good question. I think what we used in this study, what I showed you in vitro, is a non-acetylated protein because it binds both. Also the TUT mutant that is mutated in the acetylated form also binds to SIRT1 and blocks it. However, we think like you that the acetylated form might bind more avidly and might eventually be a better blocker.
Okay, we have a few more questions here. So any links between mitochondrial sirtuins, autophagy, and ER stress that you have found in your research?
We have very much looked at the mitochondrial sirtuins. I think we see also a downregulation of SIRT3 in these cells, less pronounced than with SIRT1. However, we think that this is because SIRT1 controls SIRT3 expression, and that there might be a link through this. So we are still, looking into this. I think it's a very important question. We love the mitochondrial sirtuins, so if there's a link, it would be great. However, we think it's an indirect link so far.
All right, I think I'm gonna give you one more question here because we're coming close to the clock. Have you looked at the role of any other sirtuins in
Yes, that's similar question. I think we have focused a little bit on SIRT3. We looked at sort of the expression of all of them. They're all the same. They're unchanged except for SIRT3. And we have been sort of looking into the question, what of the downstream effects is SIRT3, or what potential downstream effects of SIRT3 could be important. We think it's mainly in the metabolism, but we will have, as before, we will be very open because I would have bet my money on metabolism before, and it was not metabolism. So I'm really looking forward to finding out whether there is a role of SIRT3 and what this role is.
Excellent. Thank you so much, Melanie. Wonderful talk. Look forward to hearing more findings from this line of research in future. Now, I want to thank all the speakers, and I want to hand it over to Dr. Compton to give us some closing remarks for this well-attended Avant-Garde Symposium event.
Well, thank you, Kavi. I really want to thank all the speakers for a wonderful afternoon for those of us on the East Coast, and for a wonderful morning and early afternoon for those of you on the West Coast. That's one of the advantages of this Zoom life, is that we can join forces all across the globe in a single occasion. It really has been a tour de force set of presentations covering everything from remarkable advances in basic cell physiology through prevention, intervention development, population sciences, immunology, and finally, Dr. Ott, I'm really struck by, I'm old enough that I'm still so happy that we get to focus on aging for our AIDS and HIV patients. That was not an issue, well, it is quite a few years ago now, but it's just a wonderful change for those of us that have been in clinical medicine for a long time. And I appreciate your fascinating work looking at the comorbidity of HIV and HCV. That's a major theme, and as we see continued outbreaks of HCV around the country, frequently overlapping with HIV. This is a really important topic for NIDA and for NIH overall. I'm part of a group that's looking at eliminating HCV as a complement to the elimination of HIV that's now an international goal. So really appreciated learning about your exciting program. This is really a fantastic set of presentations and a wonderful example of what the Avant-Garde program has meant in terms of support to science through supporting the individual scientists that are represented here, and you all representing your large number of colleagues throughout this wonderful program. And with that, I'll turn it back to Kavi and Radana for any final words before we wrap up for the day.
You can go ahead, Radana.
You get to unmute.
Just thank you so much. It's been a wonderful afternoon. And I want to give a special thank you to Kavi, because she has really been the driving force and pushing to have the symposium for both the Avenir and the Avant-Garde recipients. So I really appreciate all of her dedication and commitment and the way in which she continues to want to showcase all of your all's work.
Thank you, everyone. It's been a privilege learning your work, and we look forward to hosting you again soon. And please do keep us informed. If there is any exciting finding or a manuscript in the pipeline, let us know, and we would be very proud to advertise the work and take all the credit we can. And any way we can support you too.
Thanks very much, everybody.
Thank you so much. Really cool symposium. Thank you.
Bye.
everybody.
Bye, thanks.